Devraj, and Director NRC Equines, Dr. T.K. Bhattachariya, and Dr. Niranjan Mishra, who is representing NIHSAD, Bhopal, and the directors of all animal department, and then Managing Director of Nagaland Livestock Development Board, then Dean of Veterinary College, Jaluki, Nagaland, and our RAC members, Dr. Ravindra Sharma sir, and then we have Dr. Pan sir, I can see, Dr. Arnab Sen, who is the new head of IVRI Kolkata Center. At present, he is the joint director of Nagaland Center, and many others veterinarians who have joined from different states of the Northeast, veterinary doctors, and then the scientists of NRC and Mithun. So first of all, as you all are aware, lumpy skin disease, the outbreak of LSD across India, especially in southern states, and then in Maharashtra and all, we could see it has caused havoc across, and we were getting many calls. Even when I was in NRC and Meet Hyderabad, we were getting many calls regarding it. And now this LSD outbreak has started in Northeast also. We are getting pictures from different states of Northeastern states. So in this connection, we got call from Department of Animal Husbandry, Arunachal Pradesh, that what should be done, what should be done. In that, so there, one of the options available was go for preventive vaccination using the goat pox vaccine. But when they contacted, then I apprised them that ICR is in the forefront of fighting this LSD in India. Our institutions, NIVEDI, National Institute of Veterinary Epidemiology and Disease Informatics, located in Bangalore, NIHSAD, High Security Animal Lab, and NRC Equines. They are working tirelessly towards containing this disease. They are undertaking research in identification, developing the diagnostics, and NRC Equines recently they have developed the vaccines for LSD, and even then we conveyed that, and then I also contacted NRC Equine Director Bhattachariya sir. He was happy to share some of the diseases, and also NIHSAD Director and NIVEDI Director. So now we need to discuss and we need to take decision. As of now, there are no incidences of LSD in Mithun, but it is a boss species, so we call it as boss granians. It is there. So now, but the disease is now being reported from Nagaland also, Arunachal Pradesh also. We have around 3.9 lakh population of Mithun, 90% of them in Arunachal Pradesh. So remaining are there in Nagaland, Manipur, and Mizoram. So in the census to census, if you see in the as per the 2019 census, we have seen this is the only species where 30% increase in population was seen. After every census, population is increasing. The highest growth rate, if we see in any of the species, that was Mithun. So now it is very important for us to put our brains together and chalk out the strategies how to prevent occurrence of this disease, because this is a high costly animal. Minimum each animal costs around 1 lakh rupees. So it's important for us to take some preventive measures and prepared for it. So in order to discuss this, when we approached our Assistant Director Generals, both of them, frankly, Dr. Amrish Kumar Tyagi sir, Ashok Kumar Tyagi sir, both of them were very positive, highly supportive, and they agreed to join the program. So we invited Dr. Amrish Kumar Tyagi sir to join as chief guest and give his inputs on behalf of ICR. Sir, in spite of his busy schedule, recently I had been to Delhi and I seen how busy sir was. In spite of that, looking into the importance of the LSD and looking into the importance of the livestock sector in the Northeastern region, sir agreed to participate. Sir, on behalf of all the stakeholders of Northeast and on behalf of ICAR-NRC and Mithun, we welcome you, sir. And when I requested Dr. Gulati sir, Director NIVEDI, to give a presentation on basics of this LSD, because it is helpful not only for containing in Mithun, but also for the other species, for the veterinarians of the Northeastern region. Sir readily agreed, and then he has provided, instantly agreed. I heartily welcome, Dr. Gulati sir on behalf of stakeholders of the Northeast and on behalf of NRC and Mithun. And when I approached Dr. T.K. Bhattacharya, sir, he told that there is no need to go for goat pass vaccine. We already have LSD vaccine available, and he was ready to share some of the vaccine for our people, provided we write the letters and proper communication is done. And sir also readily accepted to come and share his experiences in the vaccine production. I welcome Dr. Bhattacharya, sir. I also, Dr. Aniket Sanyal, sir, Director NIHSAD, he was also positive, but he had some overlapping program, another meeting, so he deputed Dr. Niranjan Mishra. He is a principal scientist from NIHSAD. I welcome Dr. Mishra, sir, for this program. I also welcome all the directors of animal husbandry of Arunachal Pradesh. I can see Nagaland and Manipur and Mizoram, Nagaland Livestock Development Board authorities, and Dr. Arnab Sen, who is a joint director of Manipur Center, and joined Dean of Jaliki Veterinary College, Shakuntala Madam, and the staff from staff, and all the veterinarians. Some of the, I could not notice some of the names. So I welcome all of you for this program. I also welcome all the scientists. So now to begin with, what we are thinking is first there will be a presentation by Dr. Gulati, sir, on basics of and the status of LSD in India, and then we'll have a presentation on diagnostics of diagnostics for LSD by Dr. Niranjan Mishra. It will be followed by the presentation by Dr. D.K. Bhattacharya, sir, on LSD vaccine, and then we'll take remarks of different directors and directors of animal husbandry and veterinarians from different states. They can ask their doubts and take the clarifications. So this is how the program. Now, before beginning with the presentation, I request ADG, sir, Dr. Amrish Kumar Tyagi, ADG, Animal Nutrition and Physiology, ICR, to give his initial remarks.
Thank you, Dr. Patil. Thank you very much. And
You,
my friends are here. Dr. Gulati, Dr. Bhattacharya, Dr. Sen also is there. From the, I think he has joined IVRI. Dr. Arnav Sen is also here. Dr. Sarma, Dr. Pan, my seniors, all my seniors are here. And with the directors of Nagaland, from the Animal Husbandry Department, Arunachal Pradesh, Madam Dean, and all the staff and all veterinarians. Dr. Patil has selected a very good topic as related, because recently everybody knows that how much we lost, not only in the milk production, not only production, even our animal also. And recently the milk production was in India goes down because of this particular disease, I can say. And that the talks were going on that how we can cover up, or we have to import the milk products, all these things. Actual region, actual from where it was not known, still not known, but most of the mostly it was started from the Gujarat and Rajasthan, and Gujarat and Rajasthan even more affected, even as compared to Gujarat. I can say more than total more than 1 lakh animal was dead during the 3 months from July to September, I think so. And because of that, and out of 1 lakh, almost 50,000 were reported from Rajasthan only. You can see. So how much lost the animals in Rajasthan. And
I
then
think that when you join, no, that option was there.
And then it went to the Odisha and other state also. We still could not find out the origin of the source of the disease in India, where when it outbreak in 2022. But initially also it was there in 2019 and 2020 also it was there, but not this much loss was there. And, I'm thankful to our IVRI Institute and other institute who started the work on the lumpy the skin vaccine, even the vaccine, even the our Equines Institute also have worked very hard for that particular. And starting, we took the measurement and we started with the goat pox vaccine, which was also found most effectively in this case. No doubt, later on LSD also we got, and it really effect, and we saved the animals. And we are coping up with the milk production and all these things. And this disease was mostly found in the cattle, but as Dr. Patil told that maybe it affected to the other host species. So no doubt this disease may be affected to the Mithun and yak. I don't know much about this. Dr. Gulati and Dr. Bhattachariya will tell better than me all these things. But I'm thank you to Dr. Patil who allow me to join this important topic. And I'm thank you to everyone from the Mithun Institute. I have been there, Mithun Institute, and really, Dr. Patil, I can say that you will enjoy the institute. And I have been there 2 times. There you will find a better team. All facilities are there. Animals are there. In labs are there. The only thing that we have to take some start. When Dr. Vijit Mitra also was there, he also started some work and it was going well. Another team, Dr. Haq is there. I think Dr. Khan is already left, as I discussed with you. And many 2-3 scientists are going to join, 3 scientists are going to join there. So you can make one team, good team there. And regarding this disease, no doubt better discussion can be done by the Dr. Gulati and Dr. Bhattachariya. So once again, thank you very much. And my regards to my seniors, Dr. Pan, Dr. Sharma, Dr. Annam Sen, and to all. And I think we will join very soon in August, September, when RSC is going to be held in September, Dr. Patil.
Yes,
So
sir.
thank you very much to give me chance to join with you.
Yes, sir.
Thank you very much.
Thank you, sir. Thank you so much. And as you told that we from 20th to 24th, we have kept RAC meeting, and recently we have submitted. So kindly block your dates and join us, sir. And we can also visit our conservation unit and regional center. As sir told, I think the best way forward is prevention only, because there is no treatment for this disease, for any, in fact, any viral disease. The major disease for Mithun is FMD. Now we don't want any other new disease to come, especially LSD. So it is very pertinent to have expert opinion on this issue, the scientists and researchers who are working in this area. So I request Dr. Baldev Gulati, sir, to give his presentation on LSD.
Thank you, Dr. Patil, for giving me the opportunity to interact with you people. Will I be able to share my screen?
Yes, sir.
Is my screen
visible, sir? Visible, sir.
visible? So, Honorable Dr. Tyagi sir, the ADG, Dr. Ravinder Sharma ji, Dr. Patil, and all the dignitaries present in this meeting. I'm asked to talk about the status of the mitigation strategies. So during last few years, especially the last 1 decade, we have faced many challenges to the livestock sector. These challenges are in the form of emergence of transboundary animal diseases, which have significant health and financial consequences. Some of the diseases I have listed here. In 2011, CCHF came in India. 2013, porcine respiratory reproductive syndrome came. Then 2019, we had the Lumpy Skin Disease outbreak. 2020, we got African swine fever. 2021, we got monkey pox. So We,
Benji, you have muted everyone or what?
So request all to mute themselves. Sir, Gulati sir, आप ही mute पे हैं.
so now,
Yeah.
am I audible?
Audible sir, audible sir.
So during last 1 decade, many transboundary animal diseases have emerged in India, and we are faced with the situation where we have inadequate capabilities and interventions for infection prevention and control of these diseases. So we do not have any response systems, and we faced these outbreaks, including COVID-19 in 2020. So why these diseases are emerging? And what are these transboundary animal diseases? These are highly contagious or transmissible diseases, have the potential for very rapid spread, irrespective of national borders, not only considering the state borders, and they cause serious economic and possibly public health consequences. So these transboundary animal diseases, one of them has been the lumpy skin disease. So my talk today will be focused on lumpy skin disease, and I will be talking about how this disease, what is this disease, and how this disease emerged in India, how it spread to different states in India, what is the current status of the disease, and what is the impact of lumpy skin disease, including the economic impact, and what are primarily prevention and mitigation strategies for this disease, and what steps we have taken as a government of India or at different levels, what steps we have taken. I will be talking on those also. So lumpy skin disease, whenever we talk of any livestock disease, or for that matter, any disease, we must be knowing what is the case definition. So coming to lumpy skin disease, when we talk of the case definition, we talk of the suspected case as well as the confirmed case, or sometimes we also talk about the probable case. So suspected LSD case is one with fever, lacrimation, nasal discharge, hypersalivation, followed by characteristic eruption on the skin, which we call as lump, and other parts of the body. In 50% of the susceptible cattle, there are enlarged lymph nodes, and these nodules on the skin are well circumscribed, round, slightly raised, firm, and painful, and they may be from 0.5 to 5 centimeter in size. And also we find in some animals pneumonia. So you can see in the picture on the right side, the lumps or the nodules are shown on the skin of different livestock. And lumpy skin disease is called to be confirmed case if it is satisfy the clinical case definition as above, and it is confirmed by laboratory diagnosis, then we call it as a confirmed case of lumpy skin disease. So coming to how this disease is transmitted. It is primarily transmitted by the vectors, insect vectors, either by infected ticks, infected mosquitoes, infected flies. These flies from infected animals pick up the virus and transmit to the susceptible animals. And this is possible in short distance. Because vectors cannot go long distance, the virus can also be transmitted through injections or iatrogenic routes. But for long route transmission, long distance transmission, some mobility through vehicles is also required, either the transfer of animals from one area to another area, or transfer of vectors from one area to another area or another state or another boundary through porous borders or porous state borders. This movement, where there are no restrictions of the animal's movement, this disease can be transmitted to different states. When this virus infects the animal, it produces characteristic lesion in the infected animals, which include typical gunshot wounds in the respiratory system, focal pneumonic lesion on the lungs, multiple focal necrotic foci on the liver, elevated ulcerative necrotic lesion in the abomasum, gallbladder, tongue, in addition to what we find the typical nodules on the skin. or the cutaneous form. So whenever we talk of a disease, we always talk of the triangle of the epidemiology or epidemiological triad. So when we talk of the lumpy skin disease, it is not only the virus, the whole pathogen, the agent which is responsible for the outcome of the disease. It is also, in addition to the virus, the host factors and environmental factors that play in the consequences of the infection in the animals. So regarding talking about the virus, the agent factors, lumpy skin disease virus is a pox virus, and it is single serotype. When I say single, although this belongs to Capripox virus genus, where there are two other related viruses, which we call it sheeppox virus and goatpox virus. So these three are related viruses. They are related in terms of antigenicity, immune response, and even in protection, cross protection. So, but for lumpy skin disease, there is no much of antigenic variation. It's single serotype. And this virus can remain stable in blood for 5 to 16 days, in the skin nodules up to 39 days, in the scabs for several years, even in the semen, virus is shed for up to 42 days. And this virus is transmissible through the vectors, which we call as flies or other ticks. There are 35 hematophobia flies and 17 out of 19 ticks that we have tested were found to be positive for lumpy skin disease by PCR. So there are different varieties of insects involved in transmission of this disease. Then coming to the environmental factor, virus is relatively stable but inactivated by heat, sensitive to acidic and alkaline pH, and vectors play the important role in the environment for transmission from infected animal to the susceptible animals. Coming to the host, the cattle is the most susceptible animal, cow, rather than buffaloes, although disease has also been reported from buffaloes. But if 100 cattle are infected, only probably one buffalo might be infected. They are relatively more resistant to lumpy skin disease as compared to the cattle. But within cows, very young animals are more susceptible. Crossbred animals are more susceptible as compared to the indigenous animals. But animals which are properly fed or which are relatively taken care of, better nutritional support is there, they are getting less severe disease as compared to the indigenous animals which are kept in gaushalas where there is lack of proper nutrients. So their immune system is also weak and they get more disease in the form of lumpy skin nodules. Coming to how this virus came into India, this lumpy skin disease was reported in March 2019 from Russia. Then it entered in Israel in June 2019. Then it was reported from Israel and Russia in July. And in August, it was also reported from India. So first case of lumpy skin disease was reported in August in India in 2019. And we suspect that disease entered from the neighboring countries, either China or Nepal or Myanmar. And the population which is at risk include the cattle population, the entire 192.5 million cow population and 109.85 million buffaloes populations. So we don't have any information about how much of the 3.9 lakh Mithuns are susceptible or whether they are susceptible or not. Dr. Patel will be providing that feedback. But these cattle and buffalo population are the population at risk for lumpy skin disease, have been at risk. So from Orissa, first case of this lumpy skin disease was reported. And then subsequently from July to December, the disease spread from Orissa toward eastern side. It was reported from Assam and also to western side, West Bengal and Telangana. So disease during this up to 2019 was reported from Orissa, Assam, West Bengal and Telangana. Then in 2020, disease further spread from Telangana to down south. It reached to Tamil Nadu, Kerala, and also western side, Maharashtra and Karnataka, and also from Assam, it entered in Arunachal Pradesh. And then from July 2020 to December 2020, it further spread from Maharashtra to Madhya Pradesh and also to North Karnataka. So disease has been moving relatively faster. So by December 2022, the disease was reported from more than 24 states of India, and there were more than 29,26,039 cases by December 2022 reported from India. So if we come to the spatial pattern of lumpy skin disease, during 2019, initially reported from Orissa, which we call as first wave of the lumpy skin disease, because COVID-19 disease gave the new terminology called the waves. So lumpy skin disease also we try to use these words. So during first wave, there was significantly high morbidity, but there was very low mortality. And disease was primarily reported in the form of cutaneous or the dermatitis. The nodules were there. There was no respiratory tract involvement or no generalized involvement, and mortality was very low during 2019. So that is why probably this disease was not taken very seriously by different stakeholders. From 2019 to 2021, which we call a second wave, again, morbidity was very high. It was higher than the first wave. Mortality also increased. The disease was, in addition to cutaneous or dermatitis form, respiratory form was also reported. During year 2022, which we call as the third wave, it was the most severe wave of the lumpy skin disease. Very high morbidity, very high mortality, and disease was in the form of generalized. Not only cutaneous and respiratory form, it was observed from different organs. Virus was present everywhere within the animal and outside the animal. So if we come to the number of cases or outbreaks and attacks during different years, 2019, there were only 641 cases. During 2020, there were more than 2 lakh cases and just 11 or 12 deaths reported. During 21, there were more than 86,000 cases and 16 deaths. But 2022 was the very severe for our bovine or livestock. There were more than 23,85,000 cases and more than 1,28,603 deaths reported. And this disease even continued during 2023. We have reported more than 34,000 cases during 2023 as of May 23, and 3,289 cases of deaths due to lumpy skin disease during 2023. So if we look at the state-wise distribution of lumpy skin disease, the most severest states affected were Rajasthan, more than 16 lakh cases, Karnataka, more than 3.5 lakh cases, Himachal Pradesh, Maharashtra, Punjab, Madhya Pradesh, Haryana. So these were the states which were most severely affected by the lumpy skin disease. Although there were also the eastern states like Sikkim and Assam or Arunachal Pradesh, the disease was reported from these states also. But most severely affected states were the Rajasthan, the Karnataka, the Maharashtra, the Punjab, the Haryana. And overall, till December 2022, the disease was reported from 24 states. There were more than 335 districts affected due to lumpy skin disease. The population affected were more than 24 crore 45 lakh 46,000, and total population which was affected, or the cases, total cases reported were 29 lakh 26,039 till December 2022. And disease continued even in 2023. So this table shows the data of cumulative data as on 15th May 2023. The population now, during 2022, vaccination was also started. So we have the data a population at least risk as well as population that has been vaccinated. And we have till date, till 15th May, 33,61,000 cases and nearly 2 lakh deaths, which have been reported or documented by different authorities, whether the central government or the state government or the NIVEDI or other place people. They have totally reported these number of deaths. Probably the reporting might be little under-reporting. So we can see that at least these number of deaths have already occurred due to lumpy skin disease. And this is a significant number. So during 2023, although during December, after October 2022, disease subsided to some extent, maybe due to vaccination or maybe due to development of immunity post-infection. The disease subsided in many states. Number of cases, new cases reported were almost zero in some of the 24 states that reported earlier. But 2023 re-emergence of the disease in these states, we again reported disease from some of the northeastern states as well as from some of the states, Maharashtra, Karnataka, Rajasthan. Even disease was reported, severe outbreak was reported from Uttarakhand and Himachal Pradesh, J&K also, the bordering states to Nepal, we found the emergence of disease during 2023. And these were the areas, especially Uttarakhand and Himachal Pradesh. The disease was in 2023 reported from those areas where during 2022 vaccination was not done effectively or no vaccination was done. So what I mean to give a message from this picture, that vaccination was critical, and those states who implemented this vaccination effectively, mass vaccination, they were able to better control the disease, and situation is better in those states during 2023. So disease, northeastern states, although it is not primarily a bovine dominated areas, but still the disease has been reported from 4 states, including Meghalaya, Arunachal Pradesh, Sikkim, Tripura, in addition to Assam, that was reported earlier. So these are the cases reported from eastern states during 2023 and 2022. So when we talk of the seasonal distribution of lumpy skin disease, we have analyzed the data of all the 4 years of the occurrence of this disease. And we find that disease has seasonal trend. It is more commonly reported during the month of July, August, and peak is seen somewhere in September, October, and then by December it comes down. So it is directly related with the climatic or the rainfall, because the monsoon activity starts from June, July, and it is by August, it is peak. So during this time, the vector abundance also increases. The flies, the ticks, or the other vectors which are involved in transmission of lumpy skin disease, they are more predominant. So this period from June, July till November, December, that is the most critical period for the spread of lumpy skin disease. So what we get this message from this picture, that probably if we want to prevent the disease during current year in your respective states or areas, vaccinate your animals before this susceptible period starts. Vaccinate before June. Mass vaccination for lumpy skin disease. Even if you are vaccinated during previous years, go for the booster vaccination. I will be talking about that in my subsequent slides. So this slide shows, as we have been talking with the COVID-19, the reproductive number or R0. So how this, I have just done the Karnataka case study for the transmission dynamics of lumpy skin disease. So if on the right top side, if you see the graph, there are different, this graph shows the R0 values. R naught mean reproductive number, mean how many from one infected animal, how many animals can be infected. If it is more than one, the population is at risk and there is more possibility that disease will be spreading. So epidemic peaks, if you can see of the R naught values, it is shown with the time. In Karnataka, the disease was mainly started from August 2022 until December 2022. There was a peak of the disease, and R naught value was varying from 2.5 during August, September, and it came down to nearly 1 by the end of the year 2022. So, and this graph indicates that there is possibility that this R naught value will be rising again. Smaller peaks will be coming in the state of Karnataka. And that has been demonstrated by the data from 2023 January to May, that we found another peak or smaller peak of the time varying reproductive number, or the disease was re-emerging or still existing in another peak in Karnataka. Coming to the left side of the figure, the graph, the map we shown, the Karnataka, the disease has been more severe in the Belagavi district or Belgaum district in the west, and the Bellary and Koppal district in the east. So these are the two districts. The severity was the highest as compared to the districts in northern Karnataka or the southern Karnataka. The reason being that when the Karnataka state started vaccination sometime early 2022, but some of these belts, Belagavi and Bellary, their vaccination was not very effective. So during the peak of the disease, as you see, the peak was during August, September, there are more susceptible population available in these two districts. So disease was causing more havoc. Again, emphasizing the significance of vaccination. Coming to the economic losses, lumpy skin disease, as you know, how much havoc it has caused in terms of morbidity and mortality. And in addition to the mortality or the deaths, it has also caused drastic reduction in the milk yield in dairy animals. Some of the dairy farms have reported reduction in milk production, more than 60% reduction in milk production. And whether it was transient or for a few months, or in some animals, the permanent reduction in milk yield has also been reported. Then draught power of the working animal was reduced. There was decreased body condition of the animal. And then again, we say that cows or buffaloes, they are more important even after deaths. Their hides and skins, they are used even after deaths. But because of lumpy skin disease, even the skin was not available, the hides was not available for using in leather industry. Or so abortions and infertility was another direct losses to the because of lumpy skin disease. So we tried to measure the disease burden, the burden of economic burden of this lumpy skin disease. We tried to systematically study in two states, the states of Rajasthan and Gujarat. We planned a systematic surveillance of these two states. We selected 2 districts from each state, one district that was highly affected and one disease which was relatively low risk districts, and collected data from these 2 districts from each state, Rajasthan, Jaipur and Kota, and Gujarat, Jamnagar and Ahmedabad. And we collected data on different parameters, and based on these parameters, we worked out the cost of disease, lumpy skin disease in these 2 states. So this slide shows the burden of lumpy skin disease in Rajasthan. If we look at the indigenous cattle, at individual level, 27,770 rupees per indigenous cattle. And for crossbred cattle, 49,600 rupees loss was occurring at individual animal level. And if we talk at farm level, the Rajasthan have the average farm size is herd size is 4 to 5 animals. So their average total loss was more than 55,000 rupees per herd. And if we work out the projections from the more than 16 lakh cases observed in Rajasthan state alone, the total loss due to lumpy skin disease in Rajasthan, except mortality loss, works out to be 2,313 crore rupees. And that is a significant value. Similarly, if we talk about the Rajasthan, Gujarat state, we worked the similar methodology for calculating the economic loss for both the states. For Gujarat state, we worked out 1,849 crore worth of losses due to lumpy skin disease in the state of Gujarat. So if we look at these two states, more than 4,000 crore rupees of loss has been reported from these two states. And if we project it to the entire country level, probably this loss will be touching somewhere more than 1 lakh crore rupees. But this is just a projection. We are working on the figures and doing the surveillance in other states to work out the economic burden of lumpy skin disease. So what these data indicate, disease has been economically devastating to the livestock farmers, similar to the African swine fever that has been devastating to the pig industry. The lumpy skin disease has caused a significant loss to the farmers who are rearing the bovine or cattle or buffaloes. They had significant economic losses to them. So coming to what are the challenges to the involved in prevention and control of lumpy skin disease? As I said, this disease was a transboundary animal disease, and we were not prepared to address this lumpy skin disease. We did not, this disease came when we were unprepared. We did not have any vaccines that was available for the large population of 300 million, more than 300 million cattle and buffaloes. There was no vaccine available. We did not know what vaccine is required, whether booster is required, what is the timing of vaccination, and what will be the post-vaccination immunity following vaccination. So the government of India, NIHSAD, worked out what to do, and they tried to put in place the biosecurity measures, but they were too meager and too late to control the lumpy skin disease. And vaccination was the only choice. And at that time, since import of vaccination was not possible, although there were commercially attenuated vaccines, but our government of India's policy is to avoid importing live attenuated vaccine into India. So this needling vaccine was not imported into India. So rather we looked for the alternative strategy. As I said, this virus is antigenic and related to sheeppox and goatpox. So the government of India, based on the available data, opted to go for the goatpox vaccination for the entire cow population in the country. So that was the decision taken, and 2022 vaccination was started. So when this vaccination was started, then again, some of the challenges came. How to do the surveillance post-vaccination, surveillance of the clinical syndromic surveillance to study the clinical status of the disease, serological surveillance, because we do not have any assays which can differentiate between infected animals and vaccinated animals. We also need to have the virological surveillance following the vaccination because it was thought that this virus might show recombination in the field. This vaccine virus as well as the field virus might show recombination, although pox viruses are relatively stable and they do not go for frequent recombinations. And then we did not have any data on vector surveillance. Which are the vectors involved? Which are the more competent vectors in transmission of lumpy skin disease? And also there was, because lumpy skin disease has come into the country, we have now also to work on getting eradication of the lumpy skin disease, or the exit plan for lumpy skin disease eradication that has to be put into place. Then vector control is another area which can help in controlling or preventing the lumpy skin disease. So what strategies should be adopted for insect control on animals or in the environment? Vector can be either on the animal bodies, like the ticks, or the vector can be in the environment, the flies, how to control them, eliminating vector breeding sites, and then field biosafety and biosecurity, which is more important when the disease is emerging, or when, and they become again important when we are thinking of eradicating and controlling the disease. So biosafety and biosecurity measures, including cleaning and disinfection of infected premises, cleaning and disinfection of the environment, because I said viral, these scabs, they are shed in the soil or the environment, the virus remains viable in the scab for relatively years, it can still remain stable. So we need to have some means of disinfection of infected premises, and then quarantine of animals into herds. Now the new animals that after the third wave or the fourth wave, then very few animals will be having the disease. So then to prevent its spread, we need to have the quarantine facilities, or we'll need to isolate the infected animals so that they do not increase the virus load in the environment and cause the spread of the disease. So as I said, there was no lumpy vaccine available at that time. So people started working, as Dr. Patil and Dr. Tyagi sir were telling, that IVRI and NRC Equine, they worked on developing the lumpy skin disease homologous attenuated vaccine, which is called Lumpy Provac. And till that vaccine, so although this vaccine has been is licensed and is getting approval for commercialization, still it is not in the market. But so far, goatpox vaccine is being done. Goatpox vaccination has been quite has contributed quite significantly in controlling, although it does not give 100% protection. It might be giving up to 70% protection against lumpy skin disease, but it contributed significantly during 2022 in reducing the virus burden in the animals and in the environment. So we are getting many questions on the vaccines, whether following goatpox vaccination, is it contributing significantly in reduction of the disease? So in this table, I have shown that following vaccine in vaccinated animals, infection with the LSD, the risk is low. It is not giving this sterile immunity that animal will be 100% protected, but there is low risk. Then severity of disease in the, we have seen some of the vaccinated animals which were vaccinated with goatpox vaccine developed the disease, but severity of the disease was low. Duration of the disease was short in vaccinated animals. Recovery was quick. Intensity of outbreak was low, or only few cases were seen in the susceptible population. Secondary complications were low. Response to treatment was highly favorable in vaccinated animals. Effect of production was minimum. Milk productivity was minimally affected in vaccinated animal. Protection of other animals was relatively higher. Disease spread was minimal. So definitely this table shows that vaccination is contributing significantly in reducing the spread of the disease from infected animal to susceptible animal. And those vaccinated animals, they are not getting the severe disease. Even if they are getting some disease, it is very low severity. So based on this information, the NIVEDI was working with the Department of Animal Husbandry, Government of India, on working out the strategies for controlling the disease. And we also recommended that booster mass vaccination should be done in the entire country. if vaccination was done during 2022, there should be booster vaccination during 2023. And we made a calendar for the vaccination for different zones. We made about 6 zones, and that was approved by the DHD in March 2023 and circulated to the entire states. It was highly recommended by DHD that vaccination should be done as per the schedule or the time of vaccination that has been designed by the NIVEDI. And this was designed based on the monsoon trend, the risk factors involved, so that, and the vaccine availability. We talked with the companies manufacturing the vaccines, when they can supply. So this plan was, comprehensive plan was made and circulated. Even for the northeastern states, we have circulated this plan for vaccination among the cattle population during the month of May, from 1 May to first fortnight of the May 2023. Booster vaccination was recommended for these northeastern states. I don't know how much this has been done. If it is being done, I hope that this is completed during the June, so that we are relatively better protected to lumpy skin disease during this susceptible period of monsoon, or the August to December. And this slide shows the post-vaccination antibody response to lumpy skin disease. Although vaccination, this data is based on the vaccination done in 2 states, the Maharashtra as well as Karnataka. And the vaccination used in these states was the goatpox vaccine. And another thing that I want to say, that protection to lumpy skin disease is not serologically or antibody mediated primarily. Cellular immune response also play relatively important role in protection. But we collected the post-vaccination data from these 2 states from different farms, and we found that 60 to 70%, 50 to 70% of these animals at 6-month post-vaccination were having antibodies to lumpy skin disease virus. These data is this information is based on the vaccinated animals. So post-vaccination, antibody response was seen up to 6-month post-vaccination. So this does not give any, I do not want to give this message that lumpy skin disease cannot occur in animals if they are vaccinated. This is just the data of 6 months. So I can only say that following goatpox vaccination, animals were having immunity or immune response, antibody immune response for 6 months. But definitely I would recommend that booster vaccination has to be done. No complacency has to be derived from this slide. So I was talking, we have to be following the exit strategy so that we attain the freedom from lumpy skin disease. Maybe we can target freedom from lumpy skin disease by 2030. So for this, what is recommended that annual vaccination should be done for consequently 3 years. I would say not only for 2023, the vaccination is done for consequently for 3 years. Even in vaccinated animals, booster vaccination has to be done. And then following 3 years of vaccination, we will be stopping the vaccination, but not stopping the surveillance. We will be doing active surveillance, and we will be monitoring the animals for development of no disease for 3 years after vaccination is stopped, that which we call as clinical surveillance. We'll be doing for 3 years, or we will be doing at least 2 years of surveillance, which will include clinical surveillance, virological surveillance, as well as serological surveillance. And then we will be in a position to declare that we have attained the freedom from lumpy skin disease, if not from the entire country, from at least from few states, or from your state or from your zone, so that we move ahead to attaining freedom from lumpy skin disease by 2030. So some of the prevention and mitigation strategies that would be applicable to other transboundary animal diseases also. They are applicable equally to the lumpy skin disease. That we need, once we are free of the disease or we have relatively controlled the disease, we will be monitoring very strategically the entry points, the prevention of lumpy skin disease at entry points. That means we need to have the enhanced border, or what we call as port surveillance, whether it is at airport route, or whether it is road route or rail route or the sea route. The lumpy skin disease, we thought previously either it entered from China or Bhutan or Nepal or these borders. So there, and also the northeastern states are the strategically located states. Their border surveillance or port surveillance is very important. So we need to have real surveillance at the border to prevent the virus again from these entry points. Then once the virus has entered into the country, we need to work on the prevention of further spread to other districts or other states. So then we need to have the movement restriction in place, animal checkpost in place, health certificates or health vaccination certificates we must be seeking for the movement of animals when they are moving from interstate, so that we can prevent further spread of if we are doing these kind of checks at animal checkpoints or checkpost, then we will be able to prevent its further spread. Then we need to have the early detection using rapid and sensitive diagnostics. When this disease came, we were unprepared for controlling it. We were unprepared for diagnosing it. Now we are better equipped. Dr. Mishra or Dr. Sanyal will be talking about the diagnostics that we have prepared, and we are better prepared for early detection, whether clinical detection or the laboratory confirmation. So we need to collect the samples and go for immediate diagnostics. And then also we need to have the rapid response teams for the purpose of outbreak investigation. Rapid response teams can be at the state level, district level, or the local level. And whenever we report new case, we need to declare a zone which we call as infected zone, which could be up to 1 kilometer of the infected animals or infected herd. And that could be infected zone, no movement there, all animals to be tested there. And then we can also make a surveillance zone which can be up to the 10 kilometer of the infected herds or infected zone. And we must be doing regular surveillance within this 10 kilometer boundary. So these are some of the strategies which can help in preventing of the future outbreaks of lumpy skin disease. With that, I would like to conclude, and I would like to thank the audience for the patient hearing and for the organizers for giving me the opportunity to talk about the lumpy skin disease. If there are questions, I would like to. Should I stop sharing my slide?
Yes, sir. Yes, sir.
Mouse.
Kindly mute. Yeah. Yeah. Thank you, sir. Thank you very much for the elaborate presentation regarding the origin and spread of lumpy skin disease. Ultimately, what the presentation reflects is the effort of the ICR and the NIVEDI in sero surveillance, and you have completely tracked where it originated, how it has spread across the country, and you also told regarding the economic losses. I think it's a very good presentation to begin with, and the main issue is vector control. Especially in Mithun, no vector control is possible because they're in the forest itself. So no vector control is possible. Regarding transboundary, also there is no boundary. So it's all one, Vasudeva Kutumbakam. So all animal movement will be there. I think that also has to be considered. And I have one question prior to that. I am seeing that Vice Chancellor of Kerala Veterinary University, Dr. Sasindranath, is also chairman of our RSA joint. I welcome him. I also welcome Dr. Manish Chetli, Director CARG. And I welcome Dr. E.R. Sen, Head LPT IVRI, and Dr. Kondaya, former Director NRC Meet and now is advisor to Alana Sons. from Pondicherry, Dr. Mandal. And we also have a head of Y20 India. Now G20 programs are going on. So young 20, Y20 India, Dr. Felice Sujeria is there. I welcome them. Sir, my question is, although we will have separate question, we'll have separate question answer session. So once it is occurring, is it suitable for consumption? Because Mithun is a meat animal. Once some 1 or 2 nodules are seen, if you can you go for slaughter and its consumption so that it won't spread and also farmers will not face any loss. Is it zoonotic?
It is not a zoonotic disease. So
Yes, sir.
consuming meat, definitely it will not transmit the disease, but still recommended that meat should be properly cooked, boiled before eating the infected animals. But I'm not aware of what is the clinical picture of disease in Mithun, whether it has been reported or not. Even if it has not been reported, we should be observing the animals critically. As you said, you cannot control the vectors, you cannot control the borders, but
Yeah.
animal observations we can do, surveillance we can do, including serological surveillance. We can test the animals for antibodies to the lumpy skin disease. Even if they were infected, they did not develop the disease, but they might have developed the antibodies. So that might be this kind of checks we can keep on representative number of animals, which are especially those Mithun animals which are directly in contact with the livestock population. So for that, NIHSAD can help, NIVEDI can help in doing the serological surveillance. If we get the sample, we'll be testing them for antibodies to lumpy skin disease. And with that, we will be getting some indication whether the animals are getting infection. If they are getting infection, they are not developing disease, they might be having resistance, as buffaloes are relatively more resistant as compared to cows. So Mithun might be still more resistant, the most resistant to lumpy skin disease. Maybe we can find some basis of resistance in those animals. So that kind of studies are important. We can definitely take up. I think that addresses your question, or do you have any other part?
Yes, sir. Yes, sir. Means it can be consumed with proper cooking. That's what.
Yes,
If we are finding any nodules, it is straight away, instead of if we are keeping the animal, it will spread. If it is slaughtered and if it is consumed, then at least prevention will be a kind of stomping out with less loss to the farmer. I think that may be an option. Anyway, sir, we'll take all the inputs and then we'll make an action plan. We'll have questions from other states bit later. I think now I request Dr. Niranjan Mishra from NIHSAD to give an overview of the diagnostics, some quick presentation, please. Yeah.
Good afternoon, everybody. Am I audible?
Yes, sir. Please go ahead.
So can I share my screen?
Yeah, please. Yeah, your presentation is visible. Please go ahead. Yeah.
So good afternoon, everybody. And Dr. Gulati has given an overview of the disease, about lumpy skin disease, epidemiology, and the spread also he has talked about. So I will be focusing mostly on the diagnosis aspect, that to laboratory diagnosis. And as you all know that this was an exotic disease till 2019 May. And in 2019, June and July, there were suspected cases in Odisha, in 5 coastal districts in Odisha. Then Government of India, they formed a committee involving scientists from NIHSAD, IBRI and NIVEDI. Then the samples were brought to NIHSAD for laboratory diagnosis and confirmation through virus isolation, gene sequencing. And based on the results from NIHSAD, lumpy skin disease was notified to the OIE in November 20, 2019. So coming to the introduction, already Dr. Gulati has presented nicely. So this is a notifiable transboundary viral disease of mostly cattle, and occasionally it affects
Please
buffalo.
make it full screen. Make it full screen, please.
So this lumpy is derived from the Lumpy means pruritus and skin nodules. So the disease was first reported in Zambia in 1929, and the clinical signs, that is the most prominent clinical signs, is the generalized nodular skin nodules throughout the body or in some parts. Like in a hairy animals, it will be in less hairy parts. Besides that, you can see the morbidity rate varies from 5 to 45%, or even it can go up to 80-90%, but the mortality is usually low to moderate, less than 10%, but it may go up to 45%, as you might have seen during the cases resurgence of LSD during 2022 in India. And the severity, it varies from subclinical infection to death. Economic impact is huge, and major economic losses is due to loss in milk production, damage to skin, temporary permanent sterility of bulls. Here I want to point that this is a major problem in semen production facilities throughout India. So recently we have seen in Punjab some of the farms where this frozen semen production is taking place. So we are testing regularly. It is being transmitted it is being shed in semen for about 5 months, 5 to 6 months even in some of the bulls. So it has to be very careful. The semen should be tested before artificial insemination or frozen semen production. And this LSD emerged in India in August 2019 from Odisha. Then it was reported by OI in 2019 November. So in nutshell, this is the distribution of LSD in South Asia, East Asia, and Southeast Asia. By 2021, you can see it is earlier it was confined to the Africa, Middle East, and Eastern Europe, including Russia. Then further it entered in 2019 Russia, China, Bangladesh, and India. So now after that, this Southeast Asia, all the countries including Thailand and all Indonesia, Malaysia, all the countries now, they have been infected with the LSD by 2021. So everybody knows that this is a causal agent of the LSD is called Lumpy Skin Disease virus. It belongs to the Capripox virus.
Dr.
So
Niranjan.
other members are goatpox and sheeppox virus, and the genome,
Dr. Mishra?
Genome, yeah.
Yeah, actually, sir has already given the introduction. You can directly go to the diagnostics.
Yeah, I'm just coming next one.
Gulati sir already explained all this.
Yeah. So this genome is 151 kilobase pair. So this LSD and sheeppox virus, goatpox virus, they are high genetic identity and more than 96% similarity genetic and close antigenic similarity. So serologically, it is indistinguishable. And 9 extra genes exist in LSDV, but disrupted in goatpox and sheeppox viruses. However, in LSDV, there are phylogenetically distinct with 3 lineages. One is wild type strains, second one is vaccine strains, and this recombinant strains recently, which is circulating in the Southeast Asia. And the host range, as you know, cattle is the principal host and water buffalo occasionally. But in some countries, like in Thailand, the mortality in buffalo also is comparable to the cattle. And recent reports of natural infection in giraffe, Vietnam, there was a reported death in a zoo. And we have seen in 2022, this fast emergence of LSD in free-ranging Indian gazelle from Bikaner. And the scientists from NRC Equine also, they have provided evidence in camel. This happened in 2022 when large-scale outbreaks were going on in cattle in Rajasthan. And experimental infection, it is susceptible giraffe and impala. Clinical disease, many wildlife, the disease has been reported. Serological evidence also, wild ruminants, because you have to know the host range for the diagnosis. What is the host range? What are the samples to be collected before anybody can send the samples for diagnosis? And the epidemiology of LSD in wild ruminants not definitively known, and it is not zoonotic. LSD is not zoonotic. So coming to the laboratory diagnosis of lumpy skin disease. So as per the World Organization of Animal Health, these are the prescribed diagnostic tests. So first one is the virus isolation or agent identification. There are either you can go for virus isolation or viral genome detection. So viral genome detection is the most commonly used technique because you can detect it very quickly. And but virus isolation is the gold standard test for fast identification in a country. Like India, first reported LSD based on the isolation of the virus from Hisar. After that, our country could report. Otherwise, only on the basis of this molecular test, you cannot report in a country. So this virus isolation is the gold standard test. So coming for the viral genome detection also, there are several PCR recommended primers have been given by the Y. So target is the P32 gene, which is the conserved gene, and many forms of real-time PCR, because that is the most quickest tools can be applied, because and it gives a very specific, because sometimes you get a cross contamination in PCR, traditional PCR, whereas in a real-time PCR that is avoided. So we conduct here all the mostly pan-Caprifax Stackman PCR and LSDB specific, wild types specific, because there are different strains. Again, they have to be differentiated. So first we find out whether it belongs to the LSDB, then whether it belongs to the wild type strain. Because these vaccine strains, they are not notifiable to the Y, whereas the wild type strains are notifiable. And these recombinant strains which are circulating in throughout the Southeast Asian countries, still Y has not considered that whether they are reportable or not. So they are in a dilemma. So in coming to the serology, virus neutralization test is the gold standard test for declaring whether it is due to LSDB, because it can cross-react with the goat pox and sheep pox. Indirect ELISA or other ELISA, commercial ELISA, they can give a tentative indication that regarding the serological status, but final confirmation has to be done by the virus neutralization test. So coming to the sampling guidelines, because already we have been involved in this diagnosis of LSD since 2019, and we have provided the guidelines to Government of India, which has been circulated to almost all the states and union territories. And this is available also at the website of the NIHSAD. So these are the sampling guidelines for LSD diagnosis, preferred samples, skin tissue or scab, but mostly what we are seeing, nasal swab and ocular swab. So they are the most important samples, because many times now the scab formation takes a lot of time. So anybody suspected for LSD, they should prefer nasal swab, ocular swab along with the blood. And if it is bull semen, because what we have observed during our last 3 to 4 years, in chances of getting detection in blood is less compared to the nasal swab or ocular swab. So they are the most preferable samples, followed by the, and of course, skin tissue or scab is the best sample. And whole blood without EDTA for serology. Skin tissue is the best source for the virus isolation. And also sometimes from nasal swab and ocular swab. And semen from bulls collect, it has to be sent through liquid nitrogen containers or -80. It has to be transported to the laboratory. These are some of the laboratory diagnoses we are undertaking. This was the first picture when we conducted in 2019. Again, there should be a differential diagnosis. As you might be knowing, there are several other pox viral diseases or related virus diseases which can confuse with LSD. There is a pseudo lumpy skin disease. So, but this you will not get a nodular lesions like LSD, but we get a lot of samples which turn out to be pseudo lumpy skin disease, not lumpy skin. Similarly, buffalo pox. In buffaloes, in India, it has been buffalo pox occurrence has been reported. So along with the buffalo pox, if some mild cases of LSD, that creates confusion. Cowpox, pseudo cowpox, bovine papular stomatitis virus infection. So all these can create confusion with the LSD. So these are some of the picture of the virus isolation and identification of the lumpy skin disease virus. And what is the current strategies in India? Because as for our guidelines, and we have provided as per the DIHSAD guidelines, ICAR-NIVEDI now is the apex national laboratory for LSD diagnosis. Since emergence of LSD in India in mid-2019, we provide LSD diagnostic PCR, diagnostic qPCR protocols to DIHSAD authorized LSD testing laboratories. Now there are almost 32 authorized laboratories across various states and RDDLs. And once LSD is confirmed, control measures are taken by the states and union territories as per the DHD guidelines. Like now, as per the current, till now, the goatpox vaccination using the Uttarakhand strain. Total states and united territories affected by LSD till March 2023 is 26. And recently also we confirmed LSD cases in Meghalaya. So that is after March. Because earlier Northeast cases, few cases we diagnosed in from Assam and Sikkim, Arunachal Pradesh. But this time we got from Meghalaya, and now also in Sikkim also it is many cases are occurring. So you have to be, because we are not getting samples from all the states of the Northeast, only few states, but the disease is occurring in all the states as per the unofficial reports. And everybody is aware the devastating LSD outbreaks in summer 2022 in northern and western Indian states, affecting millions of dairy farmers, more than 3.3 million cattle affected as per the DHD official data, more than 1,85,000 reported cattle deaths. So I will tell about what are these circulating strains currently globally. So LSDB strains, they can be classified into wild type strains, 2 lineages, and recombinant strains, there are 5 lineages, and vaccine strains. So you can see how many lineages, but antigenically they are very closely related, only one serotype. And among the wild type strains, there are 2 different lineages, ancestral Kenyan strains and contemporary Middle East or European strains. Actually, what we got in the first outbreak in India, that was caused by the ancestral Kenyan strains, not due to the contemporary, this Middle East and European strains. So that was a very, it was surprising how the virus, which was not circulating after 1960, how it has come. So that's the origin still remains a mystery, but it is suspected that some vaccines, spurious vaccine contaminated with LSDV, either with goatpox vaccine, it might have entered, or due to the movement of the animals, infected animals from Africa to India. But that is unlikely because we do not import any cattle. So still we have to find out from where the disease occurred. And currently, globally, current field outbreaks caused by wild type strains and recombinant strains till 2019, outbreaks mostly due to this Middle East European wild type strains and occasional recombinant strains in Kazakhstan and Russia. But in 2022, in LSDV circulating in South Asia like India, Bangladesh, Nepal, Myanmar, they are similar. All this Kenyan strain, ancestral Kenyan strain is circulating in India, Bangladesh, Nepal, Myanmar. Whereas in China and whole of Southeast Asia, recombinant strains are circulating. So we can clearly tell that this has not come from China, because we are having already tendency to tell that it has come through borders and other places, but we have to be very careful because of these evidences. And China and the whole of Southeast Asia, there different viruses are circulating. Whereas in Middle East and Africa, this contemporary Middle East and European strains circulating. And coming to the strains from India, already I have told this belongs to this. We have done the full genome, full length GPCR, RPO30, P32, several genes, and we have done the phylogenetic analysis. So I will see the next picture. So this is the first 2019 we got this subgroup 2 lineage, which is the Kenyan wild type strain. Whereas in Rajasthan, this 2022, we now got the evidence that this Middle Eastern lineage virus has emerged also in 2022. It has entered. So now we are having both the lineages circulating in India, though it may be more Kenyan strains and less. We have identified this Middle Eastern lineage recently in Indian gazelle, that is called Sinkara in local from Bikaner, as well as in cattle Rajasthan. And we got this also in Himachal Pradesh also. So this another lineage of wild type strain has already entered into the India in 2022. So this 19 to 21 outbreaks, the same virus was circulating, Kenyan wild type strains. So already I have told this in 2022, there is another insurgence of this wild type strain to India. and emergence of LSD in free-ranging Indian gazelle. And of course, the virus is identical to the cattle. So the cattle is the principal host. So whenever any wildlife or other bus species, suppose yaks or mithuns, so when they are sharing habitats with cattle, that is the principal mode of transmission through the vectors. So you have to minimize the contact between the habitats, share of the habitats. So wherever these cattle cases, LSD cases are detected in cattle, so those cattle and this other susceptible population, they should be kept far so that this vector transmission to be minimized. So that is the one method of minimizing these contacts. So I'll come to what are the challenges in LSD diagnosis. Already I have told there are several challenges. Current diagnostic tools fails to differentiate between wild type strains and recombinant strains, because these recombinant strains have been generated due to the recombination of the Mithun type vaccine virus and wild type virus. So it is having a mosaic of both viruses. So it is very difficult unless you full genome sequencing, you cannot tell that this is a recombinant virus. And multiple qPCR tests required for molecular DIVA test. There is no serological DIVA. It is not possible to differentiate serologically DIVA tests. DIVA-based serological are not available due to high antigenic relationship between capipox viruses, which are measured producing immunogenic proteins. And most of the states and UT have limitation in sample collection and dispatch to testing labs at the proper time. And differential diagnosis, as I told already, with Lumpy Skin Disease, Buffalopox, cowpox, pseudocowpox, that is another challenge. So these are the challenges remain. And coming to the conclusion and future, LSD emerged in 2019 and spread in several states. Source of origin is not clearly known, but it is certain there is an exotic source of introduction. The risk of introduction of LSD recombinant strains, there is a high chance because now we are having two lineages of wild type strains. So anytime this recombinant strain can come to India, that will be more challenging because those recombinant strains, they transmit without the help of the vectors even. Through contact also now reports are there. So because there will be a challenge for this diagnosis differentiation. So we have to be very careful for these recombinant strains entering to India. And there will be high impact on cattle production. Epidemiology and transmission dynamics need to be studied in detail. Efficacy of goat pox vaccine in cattle in field not known, although it gives partial protection. And in what we are seeing that it provides a good level of prevention mechanism. And there is a need of refinement in policy on vaccination strategies by Government of India. And first of all, and most important is there should be awareness among field vets and farmers for the lumpy skin disease. They are the first stakeholders to report the cases. Otherwise, in many states and union territories, it occurs, cases go on, nobody reports or nobody sends to laboratory for confirmation. And better implementation of prevention and control measures at field level, that is required. So thank you. I thank you, everybody.
Yeah, thank you, Dr. Mishra, for giving very focused information on the diagnostics available and different strains prevalent in different parts of the country. As you told, the last point, awareness of field vets, I think this program is for that purpose only. We have vets from all 4 states. Definitely, these cattle and Mithun are sharing habitats, but not to that extent because they generally stay in forest. But generally, there are contact points are there. And one point you told is it is reported in Gore and Batang something, no? Wild.
Yeah, in Thailand it has been reported. Thailand.
Yeah, Gore is a very close relative of Mithun is Gore. So that is why there are chances that it may also be seen here. Before going ahead to next presentation, only one question. You told there are 32 labs which have been identified. Any labs in Northeast for testing?
Yeah, the Gulati, NIHSAD, and in Kolkata also.
So in Guwahati, if we give the sample, can they do it?
Yeah, they will do. Otherwise, you can send few of the sample. Like I think in Nagaland, we have not received any sample till now. So if you are getting fast cases, better to send to NIHSAD. Otherwise, DSD again, they will tell that you send to NIHSAD.
So otherwise for serosurveillance, Nivedi, na?
One just clarification. Whenever any new cases occurring in some states, the samples should come to NIHSAD. Once we confirm in a states, so you can further samples you can send to the nearest RDDLs and other authorized laboratories by the DASD.
Okay. Thank you very much. I think questions will take after the next presentation. Now we have regarding vaccination, both Dr. Gulati sir and Dr. Mishra has told, but now we'll listen from Dr. Bhattachariya sir, NRC, Equine and IVRI. They have developed the vaccines. So regarding the LSD vaccination, they have already developed, they have commercialized it. Testing is in the final stage. And then they have already transferred it to the 4 companies. So more details regarding the vaccination, I invite Dr. Bhattacharya sir, Director NRC Equines, to share his presentation and go ahead.
Thank you, Dr. Girish.
Thank you, sir.
Respected Assistant Director General, Dr. Amrish Kumar Tyagi sir. Respected Vice Chancellor, Kerala Veterinary University. My dear colleagues, Dr. Gulati Saab, Director NIVEDI, Director NIHSAD, Dr. Sanyal sir, Director NRC Mithun, Dr. Girish, my senior colleagues, Dr. Pan sir, dear friends who are associated with animal husbandry. So, good afternoon, good evening to all of you. And today I will be sharing my views on development and commercialization of India's first lumpy skin disease vaccine, Lumpy Provac, which has been developed at NRC Equine as well as IVRI. So regarding this, just I am to share the screen.
It is visible, sir. Please go ahead.
Okay.
Yes, sir. Yes.
Okay. So already we have heard many things about lumpy skin disease, as well as their symptoms, their pathogenicity, and how it is being transmitted and how it is being detected. Now we will be discussing about that is the control measures, particularly through vaccination strategy, how to control this LSD, this an important disease for dairy animals, particularly cattle. And we have also found this LSD infection at very milder level at in buffaloes. And we have also identified this LSD, mild LSD infection in camels, horses, and deers. So there is a huge spectrum of these occurrences of this particular disease. So which necessitates us to develop some vaccination strategy to control this important, economically important disease, so that we can minimize the loss which are being incurred by the farmers due to this infection of LSD virus. So in that context, what we if we look at the genesis for development of this particular vaccine, and that I can share our views here. We received a letter from Dean Ranchi Veterinary College during the December 2019. They informed us that there are some animals which are suffering from skin disease. Possibly they may be the lumpy skin disease. And can you guide us, because this particular veterinary college, they asked us, can you guide us how to control and how to diagnose and control all this particular problem? So accordingly, that NRC Equine, the scientists, they visited that particular area of Ranchi in the Jharkhand, and they collected the samples. And then what they did, they analyzed the samples at our laboratory, as well as the whole genome sequences of that particular virus, what they isolated from the field samples, and the whole genome sequences was explored. And then they analyzed this particular virus is nothing but that is the lumpy skin disease virus. And when they compared this particular strains of lumpy skin disease virus with other reported strains, that we have found that this particular strain is very much close to Kenyan strains of LSDB, that infection. So from there, what we started that we cultured this particular virus initially in the primary goat kidney cells. Then we thought that we should develop some attenuated vaccine to control this particular disease. So for that purpose, initially we cultured in the vero cells, and we gave them many passages. Up to 50 passages we have given to these viruses, and we analyzed at different time points. We analyzed the nature and structure of this particular virus and what is happening, how the mutation is being happened in the different structural as well as other genes, and what are the mutations being happened at different stages of passages. And we analyzed thoroughly that how it is providing the attenuation of the virulences of this particular virus. And we observed that after 50 passages, we observed that this particular virus has lost completely its virulency. And when we analyzed the sequences, we found that many mutations have been present in the structural as well as other genes, other repeat sequences. So accordingly, we found that there is a huge deletion of around 801 base pair in the inverted terminal repeat region. And this particular region already it is deleted in the after 50 passage of this LSDB, this virus strain. And then possibly maybe some other mutations, they are also responsible for providing this minimizing the virulency. But this particular region, whatever we have detected that it is deleted, maybe that is one of the factor or one of the region which was maybe involved in the providing this attenuation of this particular virus. So accordingly, we characterized this particular vaccine candidate at after 50 passage, and we detected the purity of this particular vaccine candidate in the this particular stock. Then we found that this particular virus strain, whatever we have accumulated, it was free from mycoplasma species, and it was tested for extraneous agents also, and it was also sterile. We made it sterile, and we didn't find any toxic effect in the laboratory animals we studied. The efficacy of this vaccine candidate, we didn't find any toxic effect in the laboratory animals. And we also observed the antibody response in the rabbits as well as mice. So from there, we concluded that this particular passage, after 50 passage of this particular virus, this particular pathogenic virus has been attenuated and has lost its virulency. And this particular candidate, actually after that, we filed for the patent for that. And then we evaluated the safety of this particular vaccine candidate, whatever we have developed after 50 passage. And we found that inflammatory responses where that inoculation was made, there was no inflammatory responses. Then localized skin nodules, we didn't find any skin nodules. And then the generalized skin nodules also were also absent, but that fever was little bit of fever in some animals, very few animals we detected. That viremia was absent. Then virus shedding, particularly in the fecal sample, nasal discharge, ocular route, ocular discharge, we didn't find any virus shedding. That anorexia, then lymph node swelling, weakness were not there in the animals, vaccinated animals. Then we also analyzed the blood biochemical as well as hematological parameters. And what we found that WBC, RBC, platelets, those were more or less similar and comparable to the control animals. and that MCV, mean corpuscular volume, as well as mean hematocrit values, those were actually more or less similar to the control animals. So from there, from this hematological study, we may conclude that this particular vaccine strains, whatever we have developed at our laboratory, that is having very effect, that is very much effective against this particular disease, as well as this particular vaccine candidate is very much safe, and it was not causing any adverse effect in the vaccinated animals. Then we conducted the challenge experiment, and that challenge experiment was conducted at Mukteswar Center of IVRI, and in the calves we conducted this particular challenge experiment to explore the vaccine efficacy. And in the animals, in the vaccine we vaccinated around 10 animals, 10 calves, as well as unvaccinated under control group. It was 5 animals, it was there. And we inoculated this particular vaccine through intradermal as well as IV route, and challenge experiment was conducted 30-day post-vaccination. And what we observed that in the vaccinated animals, we didn't find any inflammatory response, whereas in the control animals, where there was no vaccination made, in those animals where challenged, this particular virus, pathogenic virus was challenged, we found the inflammatory responses. The skin nodules in the vaccinated animals were absent, whereas in the control animals, there were a lot of skin nodules, along with that generalized skin nodules. That viremia was also very much observed was present in the control animals. But fever, fever, little bit of fever, it was there in the vaccinated animals due to the multiplication of the attenuated virus particle. This is the live virus. So the possibility of rare fever we can find. But in control animals, we found that is the huge fever we observed in the control animals. So from this challenge challenge experiment, and we can conclude that this particular vaccine is very much safe if we inoculate to the animals, and also the efficacy is also very good in cattle. Then we also conducted some field trials, and around 26,940 animals we studied, out of which cattle 26,527 cattle, as well as 413 buffaloes we have analyzed for these vaccination, efficacy of the vaccination. And we also, in case of cattle, we also analyzed the pregnant cattle, buffalo cattle also we injected some vaccines, as well as lactating cattle. So in so different groups of animals, actually we have analyzed the efficacy of the vaccines, and in all the cases, those were very good, and it was the excellent vaccine as far as efficacy is concerned. And this study was conducted in different states like Jammu Kashmir, Rajasthan, Delhi, Punjab, and Uttar Pradesh. Then what that vaccine candidate we have developed here at NRC Equine, as well as that IVRI, that name we have given, it was as Lumpy Provag. And this particular formulation was single dose we have to provide, and a pack of 50 doses it is available. At our institute level. So far it is not, though it is under process of commercialization, we have transferred the technology to the 4 companies, but so far it is not available in the market because many issues are there. Before releasing any hormone, any vaccines or any drugs, we have to take permission from the DGCA, and it is already that safety experiments and all these things already it is over, and permission very shortly we'll be getting permission from this DGCA, but maybe within few months it would be available in the market. And this particular Lumpy Provag, this vaccine, that is very much safe for cattle and buffaloes of all age groups. And in the calves, in case of calves, maybe 4 to 6 months of age, we can vaccinate the calves, and one dose is sufficient through subcutaneous route, and that immunity, that minimum at 1 to 1.5 years, we can have the immunity of this live attenuated LSD vaccine, and but we will prefer to give the annual vaccination for this particular vaccine candidate. We will suggest. And in this context, we have given vaccination to the animals of NDRI farm, and they also analyzed the adverse effect of this particular vaccine, whether it is there or not. But they also reported that they didn't find any adverse effect, and there is no incidence of reoccurrences of this particular lumpy skin disease in the NDRI farm after vaccination. So we can summarize as far as these safety trials are concerned. We can summarize the whole experiment, whatever we have conducted both at the laboratory as well as field level, and we have found that the effectiveness of this vaccine is more than 99%. So it is very much safe. What we can conclude that it is very much safe and it is very much effective against the vaccine, against the strains whatever available in India. And for that purpose, we have also cross-nucleation study we have also conducted, as well as some just a minute, some cross-nucleation study also we have made. Different strains from different areas we collected, and that we found that antibodies, whatever it was induced by lumpy provac, it neutralizes all the LSD strains. So which indicates that capacity or potentiality of this particular vaccine, which has been developed at the institute. So then come to the immunogenicity study. We also conducted that immunogenicity study, that cell media, and this particular vaccine provides both cell-mediated as well as humoral immunity. As far as cell-mediated immunity is concerned, lymphocyte proliferation test we conducted both at the experimental level as well as field level, and we found in the vaccinated animals, the lymphocyte proliferation was significantly higher as compared to the control animals. Likewise, interferon gamma response, it was also significantly higher at vaccinated animals as compared to the control animals. So live attenuated, this particular LSD vaccines, whatever we have developed here, provides immunity up to 1 to 1.5 years, what we have observed in the experimental level. And all the details, all the data, safety data, as well as the vaccine production data, already we have published in the research journal. And if anybody wants to look at the data, they can find that all the data in this particular publication. And besides this, we have also developed that differentiation between vaccine candidate as well as field strains. So for that purpose, DIVA-based kit we have developed, and where we have detected this 801 base pair deletion in the vaccine candidate through real-time PCR kit we have developed. And through that kit, you can find out, you can differentiate both the vaccine strains as well as these field strains. Vaccine strains, you'll be finding that 173 base pair fragment, whereas in the field strain, you'll be getting this 974 base pairs fragments. So this differences of 801 base pair, that is actually we are using to differentiate the vaccine as well as field strains. This is very much effective when this particular vaccine would be available in the market. After that, we have to do this sort of ceremony, this differentiation between this vaccine as well as field strains. And so this particular vaccine already released by our Honorable Agriculture Minister during last year. And so far we have produced 2.26 lakhs doses, and we have supplied many doses to different states, particularly Rajasthan. We have given more than 85,000 doses in Jammu, Punjab, Haryana, Delhi, UP, Bihar, Manipur, as well as Andaman and Nicobar Island. And we are also, we have also done some seromonitoring study, around 1,981 samples we have analyzed, and we have given the report to the IVRI. Similarly, that RT-PCR based diagnosis also we have made at our laboratory, around 159 and these doses, this particular diagnosis we have made. Finally, we can we conclude that India's first lumpy skin disease vaccine was successfully developed and commercialized to the different companies. Particularly, we have commercialized this particular vaccine candidates to the four companies. One is Biovet, another one is Hisar Biosciences, third one is Indian Immunologicals, and fourth one is government of Maharashtra. Last year, Maharashtra state, they have procured this particular technology from us, and this year recently that Hisar Biosciences, they have procured this particular technology from us, and this particular lumpy Provac is very much safer than other existing LSD vaccines being used all over the world. And at experimental as well as field trials, we found that more than 99.9% efficacy of this particular vaccine. And this particular vaccine, shortly it will be available to the market. And this lumpy Provac vaccine, which has been developed by us, by using LSD virus, is much better efficacy as compared to the goatpox vaccine, what actually previously that different departments they were using to control this particular disease. Though it is having little bit some efficacies, but not complete efficacies or complete this effectiveness of this goatpox vaccine we didn't find in case of LSD this disease control. So this particular homologous vaccine should be used in the region to control the lumpy skin disease in cattle in the country. So at present, we are involved in developing or fine-tuning this DBA test kit, number one. Number two, we are because this particular disease is very poorly understood disease. So we are doing some research work on understanding the antibody, humoral as well as cell-mediated immunity for this LSD infection, and then mRNA response. We are analyzing against this LSD infection as well as epigenetic control, how this LSD virus, it is replication is being happened. So that epigenetic mechanism we want to explore, because that virulency it is completely it is gone. So it is attenuated. So that epigenetic mechanism we are exploring, that what is the basic facts which are actually providing this particular attenuation after doing 50 passages. So these are the areas at present we are involved for conducting research to control this particular disease in the country. So thank you so much. And I also convey my sincere thanks to Dr. Girish for giving us opportunity to deliver that vaccine candidates, whatever we have developed here at the institute, to communicate to the different stakeholders, because it is very much important actually to control this particular disease. And for that purpose, holistic approach we have to take, and everybody should be involved in this particular huge task to control this particular disease, or to disease in the whole country. That is not the state specific subject. That is actually whole country is being involved, because maybe last year it was there in the western side and northern side. Now it is there in the northeastern states. So in South India also they are also affected due to this particular disease. So I urge all the stakeholders, all the scientists who are involved in conducting research and controlling this particular disease in the country to take care, and wholeheartedly everybody should be involved. So thank you so much.
Thank you, sir. Thank you, Bhattacharya, sir, for very, crisp presentation on the vaccine, how we have developed its efficacy and other things. And my hearty congratulations for
Thank you.
developing Lumpy Provax, which is 99.99% effective.
Efficacy,
Very
yes. Yes.
hearty congratulations,
Thank
So
you.
now our request will be to undertake whatever studies you have shown is cattle and buffalo.
Yes.
Request you to undertake safety and efficacy trials in Mithun also.
Okay, no problem. Yes,
So-
already I have discussed with my scientists that we can do some experiment on the Mithun also, whether it is they are susceptible or not, we do not know actually. So that aspect we can take care.
Yeah, so we can plan, sir, and quickly we can undertake the studies.
Yes, yes, yes,
And
yes.
before that, can we only one question? Before doing the study, do you suggest to go ahead with the vaccination with this Provac, LSD Provac in Mithun, or it's a study? What is your suggestion?
My suggestion is that first we should do some laboratory study. After that, we should provide, because this is the live vaccine. This is the live virus vaccine. So
Okay.
before introducing this live virus to the animals, so we should conduct some experiment at our
Mhm.
laboratory level, whether it is really it is needed or not. If it is required, then we can go ahead. But without having any data on the laboratory experiment, my view it is that we should not go for this vaccination, mass vaccination for this Mithun or other species.
Yeah, actually, as we are studying, we got some images from some farmers wherein, as we are discussing, farmers that, some skin nodules. I have sent it to Gulati sir also. I think we have to confirm it. Is it the same thing? Some lesions are there, but we are not sure. Anyway, we'll confirm, sir. And
First, we should diagnose, then only we can go ahead.
okay. And now, because thing is, when vaccine is there, is it suggested to go ahead? But however, as I told, we'll undertake studies, sir.
Yes.
And then we'll give the serum before and after. You
Yes.
can suggest the number of animals. Accordingly, we have our form
Yes,
wherein we'll undertake these studies.
Yes.
So now, thank you so much, sir. Now, based on the inputs of all three presentations, we'll make one draft action plan and share with you. We will be happy if you can, all three, if you can share the presentations. It will be useful for us and all the field veterinarians. And we'll also try to make this recording and share it with the field vets. Now, I request, we have director animal husbandries of different states. They were listening. First, director animal husbandry of Arunachal. Dr. Taku, director animal husbandry Arunachal. Sir, are you there?
Yes, I'm here.
Yeah. Sir, welcome, sir. And now, as you, because it all started with your initiation and your inquiry regarding the vaccination in Mithun, for shall we go ahead or not, which led to this meeting. I request you to just highlight what is the LSD status in Arunachal in other species, and any other questions to the experts. Thank you.
Thank you very much.
Yeah.
From the presentation presented by different scientists, we are made to understand that Mithun is not affected by lumpy skin disease. That's very good and nice to hear that vaccinations are not necessarily to be vaccinated to Mithun. And so far, Arunachal is concerned from this disease. 9 districts has been affected from this lumpy skin diseases, and out of from 9 districts, total animal susceptible to this disease is 32,000. Out of 32,000, only 448 animals were affected, and from out of 448 affected animals, we have what death of 8 animals. And immediately after getting this outbreak of FMD, lumpy LSD, the all department staffs, including from right from contingency to officers, everybody is at their war footing and they are fighting, and to contain the further spread of the disease, all officers and accordingly, government of Arunachal Pradesh, they have also taken seriously, and Commissioner Animal Husbandry, he has issued advisory for banning of importing livestock from other states. And here also we have given a specific advisory to the 9 districts where this lumpy skin disease is spread, and all officers we are actually we have procured some pox vaccines also. More than 20,000 pox vaccines we have procured, and all these vaccines are being vaccinated in and around the affected districts. Especially these diseases are coming from our neighboring state, especially the Assam. And it is also informed that the entire Assam state is infected with this lumpy skin disease. So from this disease has come from our neighboring state. So immediately that interstate where these animals are transported has been actually officially that advisory or notice has been issued for banning of coming these animals to our states. And ring vaccination is going on. So in case just I have a query. If vaccines are available in the country, then these vaccines, how these vaccine will be procured, or whether these vaccines are to be provided by Government of India as per that of FMD vaccine which they are supplying from companies.
Actually, BioVet, possibly very shortly, they will be releasing this particular vaccine for commercial purposes. So at the DGCA approval, they are actually awaiting for the DGCA approval. And the experiment, safety experiment, whatever they suggested, it was already completed. And results also, that particular company, they have submitted to DGCA. So DGCA, after getting approval from the DGCA, that BioVet will release this particular vaccine. So this is the stage at present, whatever I know. So after
Can I add to Dr. Thakur's question also?
Yes, sir. Yes.
Actually, the central government is giving assistance to states under ICAR-NIVEDI. So there's 60-40 assistance they are providing for procuring vaccines. Vaccines are to be procured directly by the state from the manufacturer. So far, they are procuring a goat pox vaccine till the lumpy skin disease vaccine is available. And central government is providing assistance to the states for this vaccine under ICAR-NIVEDI program. So the natural state also can procure the available vaccine, whatever is available, whether LSD or goat pox vaccine, and go for it immediately. More important is not only the ring vaccination. Why don't you go for the mass vaccination of all the cattle? That will be important because ring can be broken anytime because of the lack of biosecurity measures and checkpost availability. That is practically not feasible. So go for the mass vaccinations. You have very few number of animals in your state. You're telling 32,000 animals. So go get the 32,000 or 35,000 doses and give the shot to every animal in your state. That will be more prudence to go for that. And it is only for a few years, this year and then followed by boosters 2 years. That's it. And vaccine is not very expensive. Whether you go for goat pox vaccine or even if LSD vaccine comes, that will also be not very highly priced. So go for vaccination for the entire population.
No, definitely, we will be going for mass vaccination. Actually, as I have informed you that till that, as of now, 9 districts has been reported of outbreak of this LSD. So this is the number which I have for entire states. It's a more than 4.5 lakh populations are there, and for entire population we are going to vaccinate, and we have already instructed all the officers who are working in the field, and we have already instructed, and vaccines are being procured, and it will be supplied to districts.
So my question to Dr. Aku was, when was the first case in Arunachal reported? And you have reported 448 cases. So
Yeah.
this has been during from 2019 onward, or just recent cases?
No. Recently only. Recently this year only.
Only 23 only?
23, yes, 23. Last part of 22. Last part of 22, and just it is going on. Now outbreak is reporting from parts of entire states. Actually, till today, 9 districts have reported infection of LSD.
Sir, I have a little correction here, sir.
Yeah.
Hello, I'm audible, sir. Okay,
Yeah,
sir.
please
Actually,
go
the
ahead.
first case was detected. Okay, sir. First case was actually detected in 2021, last part of 2021, in the Parsuram Kund of Lohit district. There is a Parsuram Kund. There is a farm from that animal was brought from somewhere from Gujarat and Rajasthan. That sample was given to NIVEDI, and it was gene sequence was also done. That case was detected in last part of 2020, actually 2021, in the middle of 21 and 20. After that, there was gap of within after the 20, there was a gap 22. Then after this year again May, it was erupted in this month of May.
So
Thank you.
vaccination.
Yeah. Please go ahead, sir.
So Arunachal vaccination, when was it started?
This month only.
This month only. Okay. Yes, thank you.
Sir, Bhattacharya,
sir.
sir, yeah, for goat pox vaccine also, is the efficacy study required, or for Mithun we can go directly?
For Mithun, goat pox, it is not required. You can go with, for experimental purpose, we can provide you all these vaccines for your Mithun. For experimental purpose.
Okay. That's all.
So if let us, Dr. Patil, what I would like to add,
Yes,
that
sir.
let us first confirm the status of Mithun population.
Yes.
Do we have any like you shared the picture? It looks like it can be lumpy skin disease, but let us confirm it based on sample testing or laboratory testing, wherever possible, NIVEDI or NIHSAD or wherever you want. But once we have an indication, then we can go for recommending vaccine or not vaccination. That will be second part. Without any clinical case or clinical presentation, going for vaccination right away will be difficult decision to be taken.
But the thing is, sir, if we don't go after it starts, no, very difficult to reach out to the Mithun. They're all in the forest, no?
That's why I say immediately, why not test immediately today also?
Okay, sir.
Whatever is you are finding some nodule, immediately send it to
Suspected cases.
the lab,
Collects from
get it confirmed. It will not take more than 2-3 days.
They can diagnose things. Yes.
Okay, sir. We have our farm animals. We'll collect the serum and we'll collect the vaccine, and then we'll go for
those
it.
animals showing clinical form, any nodule, we don't expect that nodules will be in throughout the body. Maybe few nodules might be there on the animals. Get 1 or 2 of them, send it to some lab and get them tested. That will be good indication.
Okay, sir. So now I request direct animal husbandry in Nagaland. Nagaland, what is his name? Nagaland. Are NLDB, officials of NLDB, Nagaland Livestock Development Board? Yeah, can you just highlight what is the status of LSD in Nagaland state? Yeah.
Good afternoon, sir.
Yeah.
I'm Dr. Gadugo. My project director is here. So I will speak on behalf of him, sir.
Yeah.
Sir, in Nagaland, LSD has not been detected for the past year, but this year we have seen some cases of LSD in 4 districts. Some of the sample we have already sent to this NIR-DDL Gulati and then ADMAC. And then we have got some positive reports of LSD from Kohima and then Dimapur. Rest of the samples are still waiting for the result. The initial outbreak was started in the month of May this year. And then we have seen the cases in the buffalo, some of the swamp buffaloes, and then in the cattle. So far, we have not received any report of LSD in the Mithun. So this is the status of LSD in our state. The department has already keeping in vigil and then given the guidelines to the public for taking the necessary action.
Are you going for any goat pox vaccination?
Sorry.
Vaccination, some of the farmers, they have already started using this goat pox vaccine.
Okay, but not from department.
Yes, sir. Department, we are yet to arrange, but from private, the farmers are arranging for vaccination, sir.
Yes.
So how many cases you have seen in LSD in Nagaland? Suspected of LSD?
Confirm laboratory confirmation, we have already confirmed from 2 districts from Kohima.
Okay, so how many cases you have seen?
Cases, sir, I
Numbers.
think total affected is below 100 yet, below 100 as of now.
Okay. Any deaths?
That report we have not seen yet, sir.
It's very less cattle is available. Below 100 but it's spreading like anything.
Okay. Okay. So I think
Can I
NIVEDI
just
has to.
give some
Yeah, please.
remarks?
Go ahead, sir. Go ahead, sir.
From Nagaland, I think we have not received any samples for confirmation. I told, as per the Government of India guidelines, LSD in a state has to be first confirmed from NIHSAD. Once it is confirmed from NIHSAD, you can send to other laboratories.
Actually, they sent to nearest Gulati, RDDL, but they should have guided actually. Yeah, they should have guided.
Sir, let me answer your queries. Sir, we are having problem of sending sample to Bhopal because the courier service are not accepting the biological sample to be transported. Even to Gulati also, we are sending in personnel, sir. In special
messenger, because we got a lot of samples from through messengers. We get from Himachal Pradesh and many places where this courier service is not reaching. Through messenger, you can send to nearest to Calcutta. From Calcutta, it is well collected. Bhopal is well collected. And second thing, once it is confirmed, it will be better for Nagaland to vaccinate all the cattle population, so that other species, they do not get affected. Because once you are protecting cattle, that's more than 90% is done. So first line of defense you should vaccinate all the cattle, because it will very less number of cattle are there in Nagaland.
Yeah.
Sir, we will try to arrange the sample to send it to your laboratory.
Yeah, okay, thanks.
Okay. So now direct animal husbandry, Manipur. Sir, are you there? Anybody from Manipur? Mizoram?
Yes, sir. Dr. Chanpuya speaking on behalf of director. Sir.
Manipur, sir?
Mizoram, sir.
Okay, okay.
Actually, we have this LSD suspected case in 2 districts recently, and we have excuse me, sir, I'll just so we have covered these 2 districts with vaccination. So the suspected samples we have submitted to NEDDRL, Gulati, and still we don't get the result so far. So the presentation we have had were very informative, and we are in full alert to combat whatever comes in respect to this disease, sir.
So in 2 districts, how many animals are affected? How many, Dr. Lalfuya, how many people are, how many animals are affected in 2 districts? You're on mute. Yeah.
Around 40, 50.
40 to 50.
Yes. Yes.
Sir, I think Gulati sir from NIVEDI, some surveillance study intensively, these 4 states have to be done. So that we'll get better information, zero surveillance. Some plan you can do, and we
Okay,
can
sir.
help you getting the samples. Yeah.
Okay, sir. Okay, sir, we'll go ahead like that.
Yeah, yeah.
Okay, sir,
Thank
we'll go ahead.
Let's see. Mute. Yeah, anyone from Manipur? They were there. Okay, there is some interconnectivity issues there. Okay. Now I request the experts, Vice Chancellor Kerala. Yeah, Dr. Sashindranath sir. Dr. Arnab Sen, are you there? Manipur?
Yeah, I'm there here. But
Yes, sir.
Manipur, the current situation is, so volatile that almost all the government offices are under curfew, But I'll just tell you 2 things. First is that in Meghalaya, the situation is really bad. There are a lot of LSD cases being encountered over the past month or so, and mortality as well. So I've got basically 2 points, because I totally agree with Dr. Mishra that this part, the northeast part, the sequencing is very important, because it may be that recombinant viruses have come over, and we have to keep an eye out, because if we look at the recent incursion of identification of 2 recombinant viruses, both these viruses had a portion of the vaccine virus in them. Incompletely attenuated vaccine virus and a wild type Kenyan virus. So that recombination led to a huge amount of resurgence of the disease. So that is one thing. And second thing is that we have to go for a very aggressive vaccination followed by a regular surveillance, because now it is not that we are past the peak. So we have a lot more to come in the near future.
Okay. Thank you, sir. If you find anything from Manipur, you can communicate immediately.
Yeah. I'll tell the director of Manipur, right?
Yes, sir. Yeah, now Jaluki madam, Shakuntala madam from Veterinary College, Nagaland.
Yeah, good evening to all. For Nagaland, Dr. Ketu is already briefed. We have a veterinary hospital here. We also get, it's a Dimapur district. Location is the Sanjivaram. Here also in the clinics, farmers are bringing few animals, cattle. For the last 1 or 2 weeks, we are getting 2-3 cases. So we have informed to the district veterinary officers, but how do we go ahead? That I'll speak with the Kedu zone. DI district disease investigation laboratory. Should we collect the sample and send it to them, or should we go directly to the NIHSAD?
So it has to
go ahead for diagnosis.
It has to go to NIHSAD, madam, as Dr. Mishra has told. First incidence state, it has to go to directly Bhopal only. After it is confirmed, it can go to Assam.
Yeah. Because we have recently we got samples from Sikkim. So I think Sikkim, it is not very far from this Nagaland. So from that, recently we got several cases confirmed from Sikkim as well as Meghalaya, like Dr. Arnab told, last 2-3 months. So all the northeastern states, they can combine and collect the samples, send the parcel with proper history as per our format. All are in the website of NIHSAD. Everything is written, how to send the samples and what are the symptoms and what are the sampling strategies. Everything is written there.
Okay.
Better fast confirm in your state from NIHSAD. Afterwards, you can send either to, better will be Gulati. Your nearest will be Gulati.
Okay, then we have all the history and all the case history. So I'll discuss with the disease investigation laboratory and I may submit to them and they can submit to the NIHSAD. That would be the Sanyal like.
No, they will not do. You have to send directly. RDDL will not do.
No. And then our Nagaland department.
As a Nagaland.
No, from college you send directly to Bhopal. No, again, when you are Nagaland.
Okay.
Actually, we
Okay,
receive
no.
some. Yeah, for clarification, we receive sample only from the government, state government.
Oh, okay.
Officially, it has to come because we report to the DISD. Because after that, control measures has to be taken seriously, and it has to be notified to the government. So it has to come from the state government.
Yeah, that's what I'll discuss with them and I'll collect the sample and send.
So NRC Mithun cannot send directly, yeah?
No, you can send with a copy to nearest the state government because they will inform, because DSD inform to the state directors.
Okay.
Director annual representative of the concerned states, not to NRC, Mithun or some other institutes.
Okay. Thank you, madam. Now, before going to other experts, I request any veterinarian from other states of Northeast, apart from these, those who have spoken, if you have any questions or queries from these 4 states or other states. Yeah. Else, I will request. Yeah, please go ahead.
Sir,
yeah.
Can I have one more question, sir?
Yes.
Last
sir.
time we had a discussion for LSD vaccine.
Yeah.
Sir, that time you had assured us to give a 50,000 LSD vaccine to the state government, Arunachal. Sir, that vaccine is for free or state government has to pay?
See, actually, when I discussed with an NRC Equines, they told they can share some few thousand samples they can share. The thing is, what is coming out of this discussion is before going that efficacy study, we should not go for it. I think that's
no,
the
sir, not with Mithun, sir, cattle population, because as you are already developed LSD vaccine for cattle. So when we are planning to do mass vaccination on cattle.
Bhattacharya, sir.
So
can you share
actually,
vaccines? Yeah.
Yes. You see, so what the government guidelines are there. In case of LSD infection, so far we do not have any LSD vaccine at the market. So whatever government guidelines are there, Government of India as well as your state government, as per the guidelines, you should go ahead. Because the Government of India possibly they have given guidance to use the goatpox vaccine. So you may go ahead with the goatpox vaccine. So far we do not have the LSD vaccine in our hand. So whatever that measures we have at present, you should go ahead with that to control. Little bit of control, because goatpox vaccine is also providing the efficacy against that LSD, that infection. So you may go ahead with that particular protocol. Whatever protocol government has suggested, you should follow that. This
okay,
is
sir. So that means we no need to wait for LSD vaccine from NRC.
Yes. If you wait for that, your animals will go. All the animals, whole animal, that population will vanish.
Okay, sir. Then we'll go with goatpox, sir.
Yes. We should protect little bit. Protect.
Okay.
Dr. Girish, if
Yes,
I may
sir.
ask a quick question. So,
Yes,
Dr. Bhattacharya and Dr. Gulati, I just need your point of view on this regarding emergency vaccination, because what is happening is that in a herd, say around 20 to 30% animals are showing the lesions for the LSD, and the other animals are exposed, but they are not coming down with the disease. So then at the state government level, there's an indecision whether to vaccinate these in-contact animals or not.
Dr. Sab, the government of India has approved so far the goatpox vaccine, number 1. Number 2, for emergency vaccine, for homologous vaccine, It is between the state government and the NRC Equine. If you request them and signed MTA that we will be vaccinating at our own risk and cost. So then I think Dr. Bhattacharya and NRC Equine will share the vaccine. But if you go, this is up to you to decide whether you want to go as per the Government of India guidelines. So follow that guideline go for goatpox vaccination. So till the approval of the Lumpy Vac Pro is obtained.
No, sir, because
If you want to go for homologous vaccine, signed an MTA with the NRC Equine and get the vaccine.
Yeah,
they are
yeah.
willing to share. I understand Dr. Bhattacharya is willing to do.
Yes, sir. Yes, sir.
right, sir. But my question is, sir, that since we don't have a lot of studies on efficacy of Capripox, because we really don't know that how Capripox, the heterologous vaccine, is performing. So whether to go for emergency vaccine using Capripox or not.
But it has been tested in our country. More than 10 crore doses have been already vaccinated.
So emergency vaccination should not be a problem, I think, isn't it, sir?
It should not be,
because
it is not.
states, they have mass vaccination they have done with the goatpox.
There is nothing like now emergency. It is already throughout India has been asked to go for mass vaccination. There is no emergency now.
What I'm saying is in contact animals which are
Yes,
not.
sir. All apparently healthy animals.
Mass vaccination. We are
Mass
going.
vaccination. All apparently healthy animals.
Dr. Manjunath
So, it can be
Reddy.
done.
Yeah.
Yes, sir.
Just
It's
now,
good.
Dr. Mishra in his presentation, he made one point. Efficacy of goatpox is not proved or something like that. He wrote, one point he has written.
Yeah, that is means no experimentally nobody has done this challenge studies and shown what is the efficacy like they have done in Lumpy Pro Vac in cattle. Although it was decided earlier by the ICAR, it was to be done at IVRI at some of the time point. But I have not come across where what is the efficacy of the goatpox vaccine in Indian cattle. There are reports in other countries. That's why when it came now in 2019, the disease came. So we decided that based on the other countries' data, they gave sufficient protection. If you inoculate 10 to the power 3.5 TCID 50, so it gives sufficient protection. So in that time we did not have anything at our hand. So that was for emergency vaccination. Government of India has recommended to use this. But this efficacy studies and further studies have not been taken seriously.
Okay. Thank you, sir. Thank you. Dr. Kondaya, sir, you had one question you messaged me. Young Kondaya, are you there?
Oh, yes, Girish.
Yes.
Thank
yes,
you very much that you are organizing a very nice event. In fact, the meat exports suffered a lot on account of this LSD and the COVID. While we rate COVID as a very successful case of control, I take it LSD is a failure of control. I request Dr. Gulati to say what is at what level the control of LSD in this country we can rate the success. See, we have nearly controlled it. It's not a very huge number of outbreaks, still they are there. Sir, why I am telling this is, to keep it in short, I have been trying for last 7 years to notify the contingency plans for some of the important diseases. The veterinary services have miserably failed, even after providing the draft, including to NIVEDI. I have provided to just vet them, improve them, and it is your duty to put it. Because when LSD came in a very limited area in the Orissa, if we have taken having a contingency plan, already a decided one, we need not waste so much time, whether we should do goatpox vaccine or sheeppox vaccine, or we should develop a new vaccine. All these things should have been avoided. Because they are already, this is a disease existing in the world, and there are adequate measures done for the control. And goatpox vaccine has been shown to have the efficacy. When you have nothing, you should go for it. So there is in the initial phase, there was a gross lacuna of controlling the disease, and with the result, it has spread to the entire country. And if any new disease like COVID comes in the livestock, what will happen? You can imagine. This is not a new disease. It is known very well. What are all the things in it? But still, the entire country got affected. So much havoc it has created. So what I want even now is, sir, please kindly get prepared the contingency plans. Now, so many questions have come here. Many of them would have answered. Even to know who are the experts of this particular disease, instead of searching at the critical moment, you will simply refer to the contingency plan. There will be a list of experts available. The states can consult them and take immediate measures. Even at the block level, we should have designated rapid action teams to inform such things. And we are not going on any very structural mode to control any new disease or any exotic or the emerging diseases if they are coming in a big way.
If anything new comes.
And why we don't have contingency plans like Australia, Ireland, or many other countries who notify the contingency plan, so that all the new entrants, all the BVSC students, all the field veterinarians, they will know A to Z of that disease. Because we discuss, the experts discuss and prepare the best information available. In that case, certainly we would have mentioned if there had been an effort of contingency plan for LSD, we should have certainly from the available literature in the initial case as an emergency measure, we should go for goat pox vaccine. If that was done in the first couple of months, it took years before we have gone for the goat pox vaccine. So, sir, I request that contingency plan for all the important diseases which are existing in the neighboring countries, we should prepare. And an unknown disease comes, what we should do should also be prepared. And who does this, whether it is the Bhopal Institute, and I take it NIVEDI should do it. And DIHSAD, I have prepared and sent in 2014 about for 6 diseases, and they asked them to send to disease experts, because I am not a disease expert, but from the available contingency plans, FAO model plans, I have prepared. So you can vet it and then get it improved and notified. We have only for, I think, avian influenza, we have one. And for one more, we have small note. That's not the one, sir. It is an elaborate one of 50-60 pages. Every information about that will be available at one place. What one should do stepwise is also available there. This is my request, sir. And I take it, it's the classical case of failure to control LSD in this country. But based on the last 3 years, what all has gone. Thank you, Girish.
Thank you, sir. Thank you. Gulati, sir. Yeah.
Dr. Saab has raised very relevant concerns, and definitely some of the points raised by him are more important. NIVEDI is playing a role in developing the action plan, national action plan. And for lumpy skin disease also, we have already made a strategic national action plan and guidelines, and we have make an action plan for eradicating this disease by 2030. It's already with the DHD. We are just the advisory body. The executing authority or implementing authority is either the DHD or the state animal husbandry. Unless we get
sir, nothing wrong, sir. Even if you put in your website a model contingency plan, let people.
Yes, the draft has already been sent to DHD,
sir, that may not
and it is waiting for their approval. And we are very right that this should be widely circulated. And in some areas, definitely India has failed in managing lumpy skin disease. It is not like bird flu or any other disease which spreads very fast, but we allowed it to spread. That is criminal, I would say, on every stakeholder involved, whether state animal husbandry or district, central or district level. We should have been more proactive in controlling this disease, but better late than never. The way we vaccinated for lumpy skin disease using goat pox vaccine, I have seen in the field the response of the vaccine, although there are no scientific studies, as Dr. Mishra was pointed out, on the efficacy of the studies, but the field data generated from these vaccination has shown that the severity of the disease, because of the goatpox vaccine or whatever vaccine, or even some of the vaccine, homologous vaccine made by NRC Equine, they have used more than 2 lakh doses, has shown quite significant. These vaccinations have significantly reduced the disease. Otherwise, 2022, there was a phase from April onward till October. Every state, there was no way out how to get out of the lumpy skin disease. But with the vaccination, we were able to control the disease. We were able to make 0 cases in more than 10 states because of the vaccination. But these efforts have to be persistent. These efforts have to be continued for next 3 years. And we have to be doing active surveillance, whether serological surveillance or syndromic surveillance, clinical surveillance, or virological surveillance, or molecular surveillance, or as Dr. Mishra was saying, or Dr. Arnav was saying, we should be monitoring also the emergence of recombinant strains. So genomic surveillance is also important. So these are the things to be taken up, not only by NIVEDI or NIHSAD or NRC, but the role of each and every stakeholder is important, whether it is state of Nagaland, whether it is state of Arunachal, whether it is state of Assam, or any state. The state animal husbandry also have the roles and responsibility. Everyone has to work as a team and contribute in attaining the mission 2030 zero LSD. So that's what I can add. Thank you.
Thank you, sir. And I think Kondaya sir's voice is industry voice, sir. He's the only person representing industry. And
very true, very true.
Yeah, hopefully you will take his. He has made some documents also earlier. Sir was telling. I think sir will contact you further on this issue. Dr. Ravindra Sharma, sir, you have former director research Hisar.
Yeah, thank you, Dr. Girish Patil.
Yes,
I compliment first Director NRC Mithun. You have taken a very good initiative, and that is as per OI guidelines, the word preparedness. Northeast, we have recently witnessed this LSD, and you have taken a right step to organize this conference, inviting Director NIVEDI, Director NIHSAD, as well as Director NRC on Equines. So it's really very good initiative. And I'm Dr. Kontai's concern is very significant. And it is as per somehow at the national level, we are missing out the basic OI guidelines for containing any kind of infections. Though FAO, WHO, why they failed in predicting corona infections or corona outbreak. But definitely now proper guidelines, they are in place. We should work according to those guidelines. And now it is not late. Dr. Gulati rightly said. Now we can work systematically to control this LSD malady from India. And for this, you need concentrated efforts. But I have got 2 queries. Yes. Excellent presentation by Mishra, Dr. Bhattachariya, Dr. Baldev Gulati. Wonderful epidemiology he is presented. And as per his epidemiology, what he's just showed, there are 3 waves. First phase, second phase, third phase. And virus is getting more vigorous, more severe with each phase. Why? Why? My question is that though we have implemented vaccination in the different phases, whether vaccination is not helping to lower down the severity of infection. And my second issue is that whether do we have any data of genetic divergence in these 3 phases of virus resurgence in the country. If we have got basically genetic divergence data of different strain in different phase, we could predict something. We could predict a model for further extension of outbreaks. So definitely we are lacking somewhere, and we need correct. We have to answer different issues pertaining to LSD. Excellent work by NRC Equines for developing our own vaccine. That's a wonderful step, and I congratulate Dr. Bhattacharya for that, and Dr. Gulati, excellent work on epidemiology. I think they should continue, but definitely what we need a concerned, what do you call, comprehensive national action plan to control this malady. And this is a pox virus, and I know as a virologist, you can control pox virus. There is not a short-lived immunity or something like that. Everybody knows it generates a CMI response very efficiently. And since this is an attenuated vaccine, so no question of low virus concentration in the animal. Even natural infection, they should. So I'm wondering why there is a R0 value. While they have, NIVEDI has calculated R0 values for different phases. And whether they have taken criteria of the surveillance, hard surveillance, antibody titers, while calculating R0 values, and the density of mosquitoes, insect density for that calculation. That will give a right picture of incidence of the disease and severity of this different phases. I think there are so many questions we have to work very hard to get some answer. So may I request Dr. Gulati if he can respond to. Thank you.
Dr. Sir, thank you very much for raising a very relevant questions, some of which can be addressed only by the scientists working on LSD. Like you said, there were 3 waves and virulence was increasing. Whether it was virulence or whether it was the other environment risk factors that were more prominent during the 2nd wave and 3rd wave, that need to be seen. As far as the vaccination is concerned, till the 3rd phase of the disease, when we found the maximum morbidity and mortality, vaccination was not implemented effectively, or it was just starting during the 3rd wave of the infection. So I don't think vaccine failure could be the reason for this. Vaccine was not playing that critical role at that time. Now it is important. Now that whatever cases we are observing, we are finding the trend that disease is now in those peripheral areas where there was no vaccination. Like I said, in Uttarakhand, we have in the disease severe outbreaks. Recently, last month also, many animals died there in those districts where there was no vaccination done. For Himachal Pradesh, similar cases, and northeastern states, now we are finding cases. These are the areas where during 2022, no vaccination has been done so far. So that itself indicates whatever vaccination has been done is playing a role in reducing the severity of the disease. You said another question, the recombination. Since so far there is only goatpox vaccination going on, and goatpox vaccine there are no reports that it is making a combination with the field lumpy skin disease virus. When this homologous vaccine comes, probably that question might arise. So far that question is not there. Goatpox vaccination was done, and lumpy skin disease virus is there. And whatever strains are circulating in India, even NIVEDI is doing sequencing. We have the viruses from 2020 wave, 2021, 22. We have sequenced and analyzing the data. NRC is also doing, probably NIHSAD is also sequencing. And in a couple of months, we'll have the real picture. We'll try to collaborate with these institutes working on sequencing. Even with Indian Institute of Sciences, we are working together. We have sequenced the strains. So far, no significant recombination events are observed within India. But the data is being analyzed, and maybe in a couple of months, we'll have good information on that aspect.
Thank you, sir. Thank you.
Yeah, vector surveillance and vector abundance is one area that has not been addressed. That's
important one. That's important one.
Yeah, and NIVEDI did some preliminary work on the hematophagous flies, and we found 17 of the 21 flies were positive for LSDB. So even tick surveillance also we have done, but it is just a preliminary. The scale of the disease, no study has been done. That's true.
Can I add something to Dr.
Yes.
Sharma's query?
Sure.
What we have observed that in the past 2019 to 2021, as everybody knows, COVID outbreak, everybody was busy in attending COVID outbreaks and other. So that time, this LSD was undermined, and many cases, because they could not do the sampling and send to various laboratories. That's why this remained unreported. Even till now, many cases are undergoing in different states and including deaths. But the field veterinarians, they fear of reporting. So this is we have to admit, this is the lack of reporting in many cases. Second one, what I have already shared in my slide. From 2019 to 2021, the same virus is circulating. Whereas in 2022, we got a different virus, lineage of virus circulating in Rajasthan as well as Himachal Pradesh. Along with that, we have shown that already the existing Kenyan ancestral strain also is circulating. But still now, we have not found any evidence of the recombinant strains which are circulating in China and Southeast Asia. And anytime they can enter. So this is the nutshell. And another point is, most of the deaths are occurring in the gosalas, cow shelters and stray animals where they are undernourished, poorly fed. In good nourished animals, the severity of the disease is less compared. So these are the three observations.
Dr. Mishra, Dr. Mishra,
yeah.
Do we have a combined vaccination, combined infections? Maybe this LSD virus is immunosuppressing the animal, and the secondary bacterial invader, they might be responsible for heavy mortality as observed in Rajasthan, Gujarat, or maybe in Punjab, Haryana.
That is possible, but very less reports are there, because unless like we have seen in BVD, because it causes immunosuppression. Diarrhea. So other diseases they clear up, but LSD is because there is a very diverse in some countries. That also depends on the breed of the animals. Like if you see in India, we are only seeing few cases of some mild infection in buffalo, whereas in Thailand and Bangladesh, lot of deaths in buffalo, though all belong to the same species. So the breeds may be different. So there are various factors, host factors, agent factors, and other factors involved in various countries playing a role, as well as the role of the vectors. Close contact.
Shall I add to that? Yes, sir. I am Dr. from NIVEDI, sir. Sir, as Sharma sir was asking, yes, there are a lot of comorbidities actually. Deaths are a lot of due to comorbid, like COVID. No, sir. We tested more than 300 samples of blood, which is LSD positive also. More than 40 to 50% are turned out to be either invariably they have high loads of hemoprotozoans.
Oh, good. Very good. This is a good finding.
Yes, sir. Either it is a Theileria orientalis, which is a new emerging Theileria in India. Actually, it causes an outbreak. So there is a comorbidity, sir. Lot of comorbidities along with your LSD. It is predisposing, many bacterial and protozoal infections.
Okay, thank you.
Any other questions from any of the participants? It's a floor
Yes,
is open. Yeah, please go ahead. Yeah.
Good evening, sir. I'm from Nagaland. I have a question, sir.
Right.
So I want to know whether the disease can infect caprine species.
So, Dr. Manjunath Reddy, any reports in goats?
Regarding LSD, sir?
Yeah.
No.
LSD.
Yes.
It is not reported. Earlier reports, they were.
No, it is goat pox is there, sir. India is endemic to goat pox.
No, LSD?
No, LSD virus cannot infect goats or sheep.
It will not infect.
No. That's why it is easier to diagnose. Any Capripax primers you can identify in cattle, that is LSD.
But field veterinarian, what he is asking, no, sir, I think he'll get definitely confused with the clinical signs, no. You will find exactly gunshot wounds, exactly the lesions similar to LSD in case of goat, but it is a goat pox actually.
Yeah, yeah.
Okay. Okay. Very good. Very good. Very good.
No, sir. Why I'm asking is because of LSD, the goat farmers, they want to vaccinate their goats with the goat pox vaccine.
Yes, it is no harm.
They can go for goat pox. That is the only solution available now. Yeah.
Yes, sir.
Any other
Okay,
questions?
okay, sir.
Thank you. Thank you. Any other questions?
Hello, sir. Can I come in? I'm Dr. Badal from Arunachal Pradesh.
Please go ahead. Yeah, Dr. Badal, welcome.
Yeah, thank you, sir. Good to see some known faces. Dr.
Yeah,
Ravindra Sharma, sir. Good to see you, sir.
Thank you, Raghavan. How are you?
I hope good, sir. Good. I hope you remember the FMD days.
Yes, yes, definitely.
Yes, sir. My question is a simple one. As you know, we are talking about vaccination and protecting our animals, like especially cattle and Mithuns. But Arunachal is home to second highest population of yak. Is there any report of outbreak of LSD in yak? Because it is also a Bos family.
Yeah.
I mean, Bos Ghanaians. So they are also, I think, as endangered as Mithun. So is there any information that I would like to know from the August gathering?
Dr. Reddy, Manjunath Reddy.
Right now, we did not receive any samples, so we did not have this one, sir. Maybe Mishra sir can.
Bhattacharya sir, shall we go for efficacy studies in yak also? What do you suggest?
Yes, we can go with for yak as well as Mithun, both under the same family, Ovidae family, and the genus also same boss. So there may have some possibility for this the receptor for this particular LSD. That we do not sure. We do not sure. We have to study actually for that.
Sir, actually yak
as well as Mithun, both. For both, we have some study.
Yak are present above 3,000 MSL, mean sea level. So far it has already reached up to 2,000. It has come. Yeah. Once it virus reaches 3,000, there are chances. So
I think winter is the best period. Yak will come down.
Dr. Patil, I think we need surveillance studies, zero surveillance in whole of the Northeast. What is the extent of LSD?
I request Dr. Manjunath Reddy with your discussion director, you should do
study with NIVEDI.
Some NIVEDI team should take some serious study in Northeast. Now you know all the issues in every state. You can make the sampling plan. Will help you to get you the samples.
Right, sir. Right, sir. Okay. From yak and Mithun, we'll design some sampling plan for zero surveillance. Then.
Yak and Mithun will let us know. We'll get from yak and Mithun, both will send it. Mithun immediately will send it. 100 must be possible immediately. So any other?
Send it. We will do it.
Yeah. Any participants? Other participants?
One more. One more addition, sir.
Yeah.
Sir, can I come in?
Yeah, please.
One more addition, sir. Whatever government of India has given as a guideline, it is basically a advisory type of thing. What we need is like a proper SOP. Proper SOP in a sense, that was prepared in when there was outbreak of FMD or during ASF, so that every state can follow according to that particular action plan. This is number one. This is very crucial. Number two, sampling. Like I say, sampling of Mithun and yak. These are also very important. So a specific SOP type of thing is need to be prepared so that the states, vulnerable states, can follow it appropriately to fit into their. This is what I think is very crucial.
Okay. Thank you, sir. Thank you so much. Dr. Pan is there?
Girish.
Yes, sir. Yes, sir.
One important thing Northeast should know is they will be all the time deficient of livestock, and there will be a movement of livestock from the other states to Northeast. So the disease risks will be generally high in Northeast. This point is an important thing, and that will be continuously there.
Yes, sir. Yes, sir. Yes. Even pigs also, almost every month around 20,000 pigs are coming to Dimapur. Yesterday, continuously we are in touch with them, and definitely other livestock, poultry, fish, everything is coming. And one more thing is, instead of telling transboundaries, in Myanmar we have many places open border. I think Myanmar in our disease control plan, it should become part of our plan actually. Whatever we do here, if we leave those open borders, there is no use. I think we should take them into confidence, and whatever livestock disease control, Myanmar and adjoining countries, small countries like Bhutan, Nepal, they should be part of our plan.
So you should maintain one good buffer zone for all the common diseases that are on the other side of the border. That is the only way then.
Buffer zone is also difficult. It's such a hilly and
no. Buffer zone means vaccinate all the animals for all the diseases that are on the other side very perfectly. So even if the disease enters in a vaccinated population, the disease outbreak won't result. It will be less than 1, the reproduction of the case. So even if one animal gets affected, it can't transfer to the other animal.
Yes, sir. Yes, sir.
So on that, so very effectively vaccinating along the border, taking a buffer zone into criteria, then the disease transmission from the other side will not enter here.
Okay. So if there are no other questions, I think we'll go for closing because already 3 hours of discussion. Any comments from scientists of NRC, Mithun? Fine. Okay. So Tyagi sir is there? Amrish Kumar Tyagi sir? ADG? No, he is not there. No? Okay. Yeah, thank you very much. And ultimately, our aim is to protect the Mithun population. As NRC and Mithun, our only concern is Mithun. And whatever the information we have collected today, we will be making a crisp document. I request all 3 presenters to share their presentation. We will make a document and we will share with you action plan for preventing, preventing and collection of samples, how to do analysis, everything, so that we will circulate it to all the Northeast states who are having. Now our request is to NIVEDI is to undertake some serosurveillance studies, both Mithun and yak, and other species. I think you have already done it. Our request to Bhattachariya sir, NRC Equines is to suggest how to go about for doing efficacy study in a quick mode, so that by the time commercially it is available, we are ready with our studies. And what is emerging is only solution available now is to go for mass goat park vaccination. Apart from that, there is no other solution. And we have to control the vectors, and we have to avoid intermingling of Mithun with the cattle. We have to see that if any, especially if there is any, you are finding that any cattle is found positive, immediately those Mithun are to be separated to see that they don't mix up. And one more action plan emerging is whenever some fresh samples are coming, first try to send it to NIHSAD, where especially in the states where there are no reports are not there. Only after sending and positive, then only you can send it to Assam, because by practice everybody sends it to AAU, Gulati, that is the RRDL. And then I think all such samples first they should go to NIHSAD, and if any support is required in sending such samples, NRC Mithun will be happy to help. And then these are the things, and ultimately all these viral diseases we all have to wait for it. They're all self-limiting. Hopefully all the viruses are self-limiting, and let us hope that it limits itself at the earliest, so that we come out of all these things. Meanwhile, we can do all these things. Thank you very much. I thank our chief guest, ADG Tyagi sir. I thank the director, Dr. Gulati sir, and his team, Dr. Manjunath Reddy, and I thank Director NRC Equines, Dr. Bhattacharya sir, and Dr. Mishra, Niranjan Mishra from Dr. Arnab Sen sir, Dr. Ravindra Sharma sir, all the directors of Northeastern States, and Dean of College of Veterinary Science, Jaluki, and all the veterinarians, and Dr. Kondaya sir from industry, and Dr. Mandal, Vice Chancellor at KASU. There are so many people I could see. Dr. Arnab Sen sir, and especially the first call came from Dr. Jimnia, Dr. Jimnia from Arunachal. He was asked by Director Animal Husbandry of Arunachal. It all started from me. So we will prepare whatever the required information we know. Accordingly, we prepare a document and we'll share it at the earliest. We also thank officials of Nagaland Livestock Development NLD. So only if you raise the issues, we can take it forward. Whenever such issues are there, kindly all ICAR institutions are working on it. Before it becomes an issue, you should go. I also thank Dr. Sangwan sir. Dr. Sangwan sir, any comments? Sorry, I missed it actually. Yeah. Unmute. You have any points? You can unmute and yeah.
Yes, Dr. Patil, thank you
Yes.
very much for enlightening all of us on LSD.
Yes,
I
sir.
am especially thankful for the three distinguished presenters, Dr. Gulati, Dr. Bhattacharya, and Dr. Mishra. One thing I want to just comment here. You mentioned about the intermingling of the Mithun and cattle be avoided. Earlier also in one of the presentation, it was said that habitat should not be shared by these two species. Actually, if we see the mode of transmission and the kind of arthropods that can transmit this disease, the range is probably very, very wide. Means the biting flies like tabanus and stomoxis, then mosquitoes, then ticks, multi-host ticks like amblyoma, like rhipicephalus. So all these, they can transmit the disease. Even if we don't allow the cattle and Mithun to intermingle, the disease will still be transmitted. So it's not, means the vaccination is the only hope for the control.
Correct.
Yeah, for this disease transmission and role of arthropods, this is, means it really complicates the preventive or control measures.
True. Yes, sir. Whereas vector is involved, intermingling is not an issue. It can go through vectors. From one.
Yes, of course.
It can easily get transmitted. Yeah.
Yes, it will get even if we separate Mithun and cattle, as your institute is also promoting fencing of the Mithun areas, isn't it?
Right.
Even
Yes,
after that fencing, due to involvement of various arthropods, the infection will be transmitted.
Yes, sir.
If you allow me, I can just.
Yeah, please go ahead. Yeah, please go ahead.
Sir, the meaning of here intermingling is when there is an infected cases of LSD, the farmer should try to separate, or it should not grazing. In that sense, it has been, sir. It's not about, because there is practically it is impractical to separate them in a grazing land or all. But when there is an infective zone, it's better that as much as possible you implement these things.
Okay. Thank you very much. And I thank all the veterinarians, directors of different Department of Animal Husbandry of all these four states who have joined. If any other issues are there anytime now, different institutions working on it, you can directly contact them. Or if you contact us, we will try to get any clarifications from them. I also thank all our scientists of NRC Mithun and the coordinator, Dr. Lalzufia, for this program. Thank you very much.
Thank you.
Thank you.
Thank you, Girish. Thank you so much.
Thank you, sir. Girish, sir, on behalf of our director, because he left to some other response, sir.
Okay.
Thank you, sir.
Thank you. Thank you, sir.
Thank you so much. Thank you.
Yeah, Bhattacharya sir and Nivedi, whenever some cattle related, buffalo related diseases you are working, quickly it should be done in Mithun also, Mithun and Yak also.
Okay. No
So
problem.
whenever you are developing something, instead of waiting till some disease reaches here,
yes,
we should do it quickly so that we are prepared. We are prepared.
Okay.
Thank you.
To work.
Yeah. Thank you, Reddy. Thank you so much. Bye.